अरे आपने ऐसे क्यों शेयर कर रखी है इसको पूरा।
हाँ जी। क्या हाल है? तुम अकेले ही आए थे क्या?
अभी तक तो मैं ही सर।
स्कूल ले लो पीछे।
पहले पूछा वो
Good morning.
गुड मॉर्निंग मैम।
Morning, Dr. Geeta. Morning, Dr. Kumar. You are on mute.
Good morning, ma'am.
Good morning. Nice to see you all.
Thank you. Thanks to you, ma'am, for giving your valuable time to us when and as required. आपको तंग करते रहते हैं हम बीच बीच में।
नहीं, that's my pleasure. Anything for profession.
थैंक यू।
You have dialed is currently busy. Please try later.
बढ़िया। मुझे है दोनों।
What is the target audience for today? Is it students or faculty?
Ma'am, students, faculty from all India, more than 1,000 participants are there. We are live on YouTube, ma'am.
Just trying to understand so that I can cater to everyone's needs.
आवाज आपने कर रखी है ना, बंद कर देता हूँ।
Sir, good morning.
Good morning.
Very good morning, ma'am. Very good morning, all of you.
गुड मॉर्निंग सर, गुड मॉर्निंग।
Namaste,
Good morning,
sir.
sir. नमस्ते सबको, नमस्कार। अच्छा, नमस्ते वंदना जी।
सर प्रणाम कर रहा हूँ।
Sir.
जी जी, सबको प्रणाम सर। जो भी लोग हैं, कटारे साहब को भी प्रणाम, और जो लोग भी हैं सबको प्रणाम।
Start करें sir?
बिल्कुल।
जी sir. Morning, everyone. I'm Dr. Priyanka Kriplani, and it's my pleasure to welcome you all for the virtual national conference on research methodology held at Guru Gobind Singh College of Pharmacy, Yamunanagar, in collaboration with Association of Registered Pharmacists, which was established to promote the interest of registered pharmacists and to advance the profession of pharmacy in the country. The conference is centered on the theme fostering research and innovation in pharmacy, advancing academics and industry collaboration. Guru Gobind Singh College of Pharmacy, it was established in 2002 with a vision that is to be center of excellence of international repute in pharmaceutical education and research. Now, moving on towards the conference, research and innovation, they are the pivotal for advancing healthcare and pharmaceutical sciences, making collaboration between the academia and industry more crucial than ever. This conference will serve as a platform to unite the researchers academicians, industry leaders, and students. And together they will explore the innovative practices and cutting edge research methodologies to drive transformative developments. It is our honor to have among us Dr. Brahmeshwar Mishra, Professor, Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, BHU Varanasi, as our guest of honor. I welcome you, sir. Dr. O.P. Katare, Professor, University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. Dr. Vandana Patrawal, Professor, Institute of Chemical Technology, Mumbai, Maharashtra. And Dr. Sudhir Bhardwaj, Director, Divine International College of Pharmacy, Gwalior, Madhya Pradesh. Dr. Kumar Gaurav, Principal, Guru Gobind Singh College of Pharmacy, Yamunanagar. I welcome you all. Now, I request our principal, sir, to welcome our guest. So, please, sir, Dr. Kumar Gaurav, sir.
Good morning, everyone. It's my privilege to welcome you all to the virtual National Conference on Research Methodology, themed Fostering Research and Innovation in Pharmacy, Advancing Academic and Industry Collaboration. This theme highlights the growing need to bridge the gap between academics, research, and its practical, real-world application within the pharmaceutical industry. Today, we are honored to have with us distinguished personality, Dr. B. Mishra. Thank you very much, sir. Our esteemed guest of honor, along with the eminent speaker, Dr. O.P. Katare, Dr. Vanda Patrawale, and Dr. Sudhir Bhardwaj. These experts have made remarkable contribution to both academia and industry, demonstrating the true value of outcome-based research. Their work provides actionable solutions that go beyond theory and emphasize the importance of tangible real-world results. While much academic research remains theoretical, the practical approaches adopted by our esteemed speakers showcase how research can directly impact the industry. Their pioneering efforts in healthcare solutions, novel delivery systems, and industrial collaboration serve as example model for us to follow. Whether it is Dr. Katare formulation for the treatment of psoriasis or Dr. Patrawale drugs eluting stents, they have successfully bridged the gap between research and industry by ensuring their findings lead to the practical application that benefit society at large. Throughout today's conference, we will explore how outcome-based research can guide towards real-world solutions, offering a clear roadmap for success. The focus is on ensuring that research not only add to the body of knowledge, but actively contribute to meaningful positive change in the world. As I continue on our research journey, let us draw inspiration from the practical result-driven work of our speakers. By following their example, we can ensure that our contribution have a lasting and positive impact on the pharmaceutical industry and beyond. Thank you, and I look forward to the insightful discussion that awaits us today. Thank you, all the speakers, for giving us time. Thank you, sir. Thank you, madam.
Thank you very much, sir. Now I take the privilege to introduce our distinguished guest of honor, Dr. Brahmeshwar Mishra, Professor, Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, BHU Varanasi. He has more than 32 years of research and teaching experience. Dr. Mishra's research focuses basically on development and evaluation of NDDS, including nanoformulations, pharmacokinetic and pharmacodynamics, and evaluation of drugs and formulations. Dr. Mishra has secured prestigious research funding from TEPTEC scheme of AICT, New Delhi, and ICMR, New Delhi. Dr. Mishra has published 190 research reviews, 195 paper abstract, and 8 chapters in books. And he has successfully guided more than 54 postgraduate students and 12 PhD students. He is also recipient of many prestigious awards. I welcome you, sir. I request you, sir, to please share your views with us. Please, sir.
To start with, I'm audible.
Yes, sir.
Yes.
I always used to offer my pranam in the lotus feet of Ma Saraswati, the goddess of knowledge. And because without the blessings of her, we can't proceed, or we can't do anything. Whatever we are today in the field of education and research, we are only with the blessings of goddess Saraswati. So my pranam goes to her lotus feet. I used to offer my second pranam always to Bharat Ratna Pandit Madan Mohan Malviya ji, who is a visionary founder of the great university Banaras Hindu University. And why I used to offer my pranam before my any talk or anywhere I am speaking is that I am since a student of also of Banaras Hindu University and I am also working there. So I personally feel that without his blessings, whatever I am today, it was not possible. So therefore, I always seek blessings of him and offer my pranam to his lotus feet. My third pranam always goes to father of pharmacy, Professor Amal Sharaf. The reason behind is that he started the pharmacy education not only first time in India, but also in first time in Southeast Asia from the department where I educated and also I am working there at the present. So therefore, and whatever we are today in the field of pharmacy is because of the blessings of him. So therefore, I offer my pranam to him. Fourth pranam here I am offering in the lotus feet of Guru Gobind Singh Ji, the Guru, because this college of
इसमें ना
pharmacy is
दो चीजें
named
देखो कि इसके
in the name
अलावा
of
हम
Guru
और क्या
Gobind.
यूज कर सकते हैं अगर
Pardon?
नेचुरली आप कर रहे हो और दूसरी बात इसमें क्या केमिकल या फाइटो कॉन्स्टिट्यूएंट है जिसकी वजह से कोएगुलेशन हो रही है इसकी एक इसके ऊपर प्लांट के ऊपर हम रिव्यू नहीं कर
तो
सकते
म्यूट करके
कम से
बाहर कर
कम
दो।
उसके 2 4 5 7 तो हो
I think I am audible.
Yes, sir.
Okay, it was looking here as a mute. Anyway,
Hmm.
so my fourth pranam goes to lotus feet of Guru Gobind Singh Ji, because I personally feel that this college is named as a Guru Gobind Singh College of Pharmacy, and it is whatever it is doing and getting the recognition in every way, every sector of pharmacy is because of his blessings. My fifth pranam goes to all the audience who are participants of this wonderful conference or webinar, whatever you can say. And in the end, I would like to thank the organizer of this one-day webinar, virtual webinar, Dr. Gaurav and Dr. Geeta ji and all others who are associated with this webinar for inviting me as a guest of honor. Before proceeding with, I will just request the organizer to, since I am feeling privileged and feeling very happy to be associated with this college at any platform very first time. Probably you have taken my CV from the my website, which is actually is not updated always. So I will add little bit into that because my experience has been now 40 years. September, I have completed my 40 years of service. 2 years more I have to still go. Publications more than 250, 32 PhDs there, more around 100 MTech MPharm. So all these. I'm sorry that updation is not there. So you can ask me whenever you call any speaker or guest, you may send a mail to send a brief one page biodata or biography. So we will give you, so that will update everything. So this is just for a kind of suggestion. Now coming to the this webinar, which is on the topic of research methodology, and it is, of course, it is not webinar, virtual national conference, and the theme is fostering research and innovation in pharmacy, advancing academics and industry collaboration. So we are discussing these issues at different platforms. Research methodology is also part of course curriculum in PhD all over India, I think. Anybody doing PhD has to clear the credits requirement for the research methodology. But here, so many things they might be already, all the researchers might be studying in the research methodology part. But as far as I am as a guest of honor is concerned in this national conference, I will just give a glimpse of certain basic things which is needed for the researcher to proceed with the research, and especially what exactly the important methods which many times they forgot or they ignore actually in doing that I will be just giving a hint, and will be a kind of suggestive or suggestions which they can take for their future research. First thing is the recognition of, that is selection of the topic of the research. I used to get the topic from the student himself or herself. I asked him or her to go in detail for the literature survey. We gave some idea that where, okay, we can work on this. But the issue is that a researcher has to go in depth literature work, literature survey, and whenever they are going to decide a topic, they have to see that what work has been done already by the other researchers all over the globe, and what work is not yet done, and accordingly one usually decide the topic. But in this process even many things are just missed, and those missing gap can be filled by seeing that you just ask the question. Let us say you have come to a conclusion to decide a topic of your research work. So then you ask that why you are going to work on this. What is the gap? The gap identification is very important, which is usually missed because of, we cannot survey the literature 100%. Some of the issues always remains, but at least we should see that. For example, I am a pharmaceutics person, so I can say that if I am going to develop a formulation of any type, so I will see that whatever the formulations which are available in the market of, then drug also comes into the picture, which drug formulation we are going to make, because drug selection is also a very important criteria for developing a formulation. So selection of drug will be first be looked upon whether it is really required to prepare another formulation, and what is the gap, what are the problems associated with the drug and the formulation which is already available in the market, and what research has been done related with this drug and the other formulations where still some lacunae is there, or still some of the things which are not getting fulfilled which is desired as far as the treatment of disease is concerned. For example, whenever we design a formulation or develop a formulation or even discover a new drug, sorry, just a minute. I'm at home and my grandson is disturbing me. Anyway, I'm sorry for that. So, therefore, selection of drug is very important. At the same time, which formulation you are going to prepare is very important, because whether you work towards the discovery of a new drug through chemical route or maybe phytochemical route, or you are working on any formulation aspect with the new development of the formulations, basically we do have three major objectives, or rather four major objectives. One is, for example, if you are discovering a new drug, you will be discovering for the purpose that let us discover a drug which should be more efficacious for the treatment of a disease as compared to existing drugs. And same is the objective for the development of the formulation as well, that if you are developing a formulation of an existing drug, So therefore, especially for the NDDS type of drug, or let us say nano formulations, if you are developing of an existing drug, the objective is to get the more efficacious result out of your formulation. So whether it is a new drug or whether it is a new formulation, research objective or the achievement is expected to be the same, that it should be having the better efficacy as compared to the existing drug or the formulation. Second objective is if you can minimize the toxicity or associated side effects or associated unwanted effects with the discovery of the drug and with the discovery of the new formulations. So second is the reduction in the toxicity, and third is overall, cost of the treatment also, if you can minimize, so that will be fine. So therefore, to achieve these three goals, what has been done till now? All the three goals have been whether achieved or not. I think we don't have any drug where we cannot explore to achieve these objectives, either as a discovery route or as a development of the formulation route. So that makes the sense of, coming to a conclusion by deciding a topic of the research that, okay, if I am going to work on this drug with this formulation, then probably the desired goal of having better efficacy with minimum toxicity and also the lower cost can be achieved, or at least one or two goals can be achieved. And of course, if you're talking about the novel drug delivery systems and the other formulations of a special type or a specialized type, so in that case, patient compliance also comes into the picture, that you can improve the compliance, you can reduce the frequency of administration, and that is also one of the great requirement as far as the development of the drugs or the formulations are concerned, towards achieving the desired goal. Then once we have decided the topic, then you will be proceeding with, and then you will be actually have to follow the different research methods you have to follow in your experimentals. Those research methodology might be involving different equipments. So you as a researcher, or anybody should as a researcher, for example, many of the times the people get done their work, which they are not having the facility at their own end, they get it done from somewhere else by hiring the services, by payment basis or even by collaboration basis. Okay, that's fine. There is no any harm into this, because if you don't have facility, you can always hire those facilities. You can get those data from getting experimented from that particular place. But the issue is, if you are generating a thesis or publication out of this work, then you must be, as a researcher, you must be aware of how to handle that equipment, how to use that equipment, what protocol of the experiment has been done, followed, how it has been done. Many times I have seen that while interacting during either selection committees or PhD vivas or many other occasions, I found that these hired services, experimental data, they are not able to explain anything. So as a researcher, you are not only person to go for the generation of a thesis for getting the degree, or for the let us say generation of certain article which is required for the getting the degree. Your also objective is to understand, to know. You are expected, everybody in the world will expect you that you have the knowledge, you have the skill of operating that particular equipment, you are completely aware of the pharmacokinetic, you are completely aware of the methodology which has been followed, each and every step which has been followed, and that is very, very important, for the researcher. I can tell you, for example, while doing the bioavailability or the pharmacokinetic studies, I have seen personally many times that if you are, let us say, estimating your drug concentration in blood, plasma, or serum of animals after dosing any particular formulation or any particular drug of which you want to measure the bioavailability or the pharmacokinetics. So what happens, the calibration curve is very important. many times the researcher use the calibration curve drawn in, let us say, PS 7.4 buffer only. So they have already in vitro dissolution or in vitro release studies, they have prepared this calibration curve for estimating, or let us say studying the in vitro drug release characteristics or in vitro drug dissolution characteristics. And the same calibration curve is used for determining the unknown drug concentration in the samples of the blood, plasma or serum which they have taken for doing the bioavailability or the pharmacokinetic studies. So it is wrong. It is, I mean, you will have to prepare a calibration curve in blood, plasma or serum in which you want to do the bioavailability studies. Similarly, if you want to do biodistribution studies in any particular organ, if your objective is to, let us say, to know that with the function of time, how much of the drug is reaching to the treatment organ or the target organ, and how much of the drug is reaching to the many other organs where toxicities are expected to be more, then we do biodistribution studies. In animals, this study can be done. And there tissues are getting extracted, I mean, taken from the animals, and those after tissue homogenizations, we are going to determine the drug concentration in those tissues or organs at different time intervals. That is known as a biodistribution studies. So in that case also, you need a calibration curve in individual organ. I am not saying that all the researchers are not preparing this. Many of them are preparing the calibration curves in individual biological samples, whether it is a blood, plasma, serum, or whether it is a organ, specific organs where they want to determine. But calibration curve is very, very important. This is a part of research methodology which has to be followed. Now the next question comes: how to prepare the calibration curve? Because in the let us say in blood, if somebody would like to prepare a drug concentration in the blood, let us say 5 microgram to 100 microgram per ml in the blood, so there is a lot of difficulty arises that how to put directly drug into the blood, or whether we have to put drug solution into the blood. So I'm not going into details of this procedure, but there are established methods are there by which you can proceed, and you can always prepare the calibration curves. So this was just an example. I think no one should take it that they are not doing it. I'm sure that many of the researchers are following this, but if someone is not following, and if they are using the calibration curve of in vitro drug dissolution studies or in vitro drug release studies, and then using that calibration curve for the bioavailability or pharmacokinetic studies, then that is not correct. So this was just an example. Because it is a conference of a research methodology, so I have to explain that methodology which are going to be followed or need to be understood is can be given by an example only. Similarly, I will say to all the drug discoverer, I mean the person who are working in the synthetic chemistry or discovering the drugs through the phytochemical route or the plant route, you all agree with me that they come out with let us say, a molecule synthesized or a phytoconstituent, if they could, let us say, standardize, which many of the times is not standardized with the different tools. Mostly they are working on the crude extract. But if it is standardized, let us say, and there is a guy, they have got a individual phytoconstituent as a new drug, or a by synthetic or phytoconstituent route, then they do biological screening simply by putting the drug into the CMC solution, carboxymethyl solution or suspension. I have seen even the drug is soluble in water, then also they will prepare the CMC solution, the CMC suspensions. But first thing is that you will have to see that whether your drug is soluble in water. They will not check it, and they will put into the DMSO also. The DMSO is a very common solvent. So my saying is that first you should see at least the physicochemical properties of your discovered drug, which you are not doing, which many of those researchers are not doing. They have discovered it, they want to do biological screening, for biological screening, let us say feeding of this discovered drug into the animals has to be done by some liquid formulation. And that liquid formulation, mostly they are either doing in DMSO or they are doing into the CMC suspension or twin AT suspension, something like that. So what I am saying is that if drug is soluble in water, you prepare the solution, and then whatever biological activity or pharmacological activity you are getting, or let us say positive things which has been achieved, or even it is a, let us say, anti-cancer drug, we have got a good efficacy into the cancer cells, killing, fine enough, don't leave it there, collaborate with someone, someone from the pharmaceutical technology or drug delivery area. Let this new molecule which has been discovered and is found to be active, let this be converted into some specific formulation, and then those formulations should also be tested in the same model where you are testing simply by putting into the DMSO or CMC suspension, because the formulation changes the effect. You may have a reduced dose for the same efficacy with the different kinds of the formulations. You may have reduced toxicity with a specific kind of the formulation. Your dose requirement may be less. Your frequency of administration may be different. So whatever conclusion after the drug discovery you are going to take, and including in your thesis or publication, that will add many times. And many times in the way that, let us say, if your simple drug with the CMC suspension is effective at 100 milligram per kg body weight level, and if you are doing in some specific formulation, you will be surprised to know that it is effective even is 20 microgram per ml level, 5 times dose reduction has been done. And this dose reduction is going to reduce the cost of the product. This dose reduction is going to, increase the discovery value 5 times more, because if 5 times dose is going to be reduced, then of course the discovery value cost is going to be improved, and also the efficacy is expected to be at a lesser dose, and toxicity is going to be very less because the dose is already reduced. So this is just my advice, that if you are still continuing with the research, don't leave the molecule discovered there only at the end of the thesis, but try to proceed with the collaborations and whatever facilities you do have. Try to understand that can we get the similar kind of efficacy or more efficacy with reduction of the dose, or with any specific type of the formulations. All these things, can be explored and can increase the value of the research manyfold. This is true for any area. This is not only true for drug discovery area and pharmacognostical phytoconstituent area, but it can be true for any area. So my humble suggestion to researcher is, and to all the participants, are that in performing all these, your honesty is very, very important. You have to be very honest researcher. You have to have the enthusiasm of understanding, knowing the skills, understanding the skills, performing the skills, and if somebody is asking, then you are able to replicate. If those experiments are performed again, you are going to get the similar kind of the results. If all these things has been achieved, because only earning the degree or having the publication is not, and having the patents are not going to serve the purpose. Once you should yourself be highly satisfied that no, I am a master or a doctor of this particular area. If somebody asks me anything to do, or ask me anything related to this particular area, I will be able to answer without any question, and I can be I should not be having any doubt in that particular case. So, ladies and gentlemen, this is I think I'm not aware how much I am expected to talk, but I think I have already talked for about 20 minutes. So this is a inaugural session. So therefore, these are some of the suggestions, and these are some of my words. I wish each and every participant that this one-day conference with the eminent speakers who are there, that Dr. Katare, Dr. Mandana ji is there, and Dr. Bhardwaj is there. They are all very well known to me, and I am very sure that with their knowledge and their presentations, you will be able to learn a lot. And whatever I have suggested is just a kind of, a teacher suggestion, which is always given to the students to proceed with the positive attitude in the research or in the academics or wherever you are working. With these words, I once again thank the organizer for the inviting me as a guest of honor and for this virtual national conference. And I wish you all very best for the wonderful conference to end. Thank you so much.
Thank you very much, sir, for sharing your valuable research experiences and practical knowledge with us. They will be definitely helpful to the teachers who are undergoing PhD or the students that are doing MPharm project over here. And BPharm students also, they are also involved in the project in the 4th year. So thank you very much, sir, for giving Thomas your time to us. We are very delighted to have you with us. Thank you very much, sir.
Thank you so much. Thank you. Can I leave now?
Yes, sir.
Okay. Okay. Thank you so much.
Okay, sir. Thank you, sir.
Thank you.
Now, I would like to introduce our first speaker of the day, that is Dr. Vandana Patrewal. She is Professor, Institute of Chemical Technology, Mumbai, Maharashtra, and has more than 30 years of teaching and research experience. She has 200 refereed publications with over 12,000 citations, 30 granted patents, 20 patents in pipeline, and 2 trademark registries. And she has also published 5 books, 45 book chapters with international publishers. She has been active in teaching, research, and services throughout her career and is listed consecutively in top 2% of world scientists in pharmacy on the basis of publications and citations from the time the list is published by Stanford University. She is also recipient of many prestigious awards and is also the member of editorial board of many peer-reviewed journals. She has transferred about 20 plus technologies to industry, including drug-eluting stents being marketed in more than 65 countries. And it's a pleasure to welcome you, ma'am. I request you, ma'am, to please take over.
Thank you, Dr. Priyanka, for those kind words. And thanks, Dr. Kumar and Dr. Geeta, for inviting me to this forum today. Can someone confirm if the slide is visible?
Yes, ma'am, it's visible.
I'll put it on the slide mode.
Yes.
Yeah, so I'll not take much time, but definitely I would like to convey to you how translational research is done. And when we say translational, I mean academic research which has a sound hypothesis and can be translated to industry. So some of the case studies will be presented. All of this work has been done at Institute of Chemical Technology, where I've been serving, and it's a real premier institute with all the facilities available in-house. And let me proceed now. And as Dr. Mishra said, that I would also like to seek blessings of Guru Gobind Singh Ji. We celebrated Jayanti just on 6th Jan. I wish this lecture was also on 6th Jan. So bench to bed key drivers, if you need to look at, they will include all these. And if you are able to satisfy each of these, that means you will have a product which will see the light of the day. That means it will reach market. So we need to start from whether that product is a necessity. Is there any unmet need for which we are making the product? Or is it just a me too kind of a product which will, not be having a business potential? Once you have done that, go to the novelty part. Is it that I can make a patent non-infringing methodology to manufacture a particular product, or if it is first in world kind of project? Both are novel. Novel, definitely people can come up with technologies, but what they do not see is whether it is feasible to scale up. So, my one major advice to everyone would be look at technologies which are very simple, which are scalable, and industry can readily take them and make it at a production scale. Obviously, anything which is novel, you need to secure IP and you need to have umbrella of patents around it. And it always helps if academicians patent their technology first and then contact industry rather than the other way around. This way, you do get definitely good royalty too. We need to also see whether we can have product life cycle management. Today you have given them some technology. For example, you have given a solution form. Tomorrow you say, okay, a solid oral dosage form is available. That also can be formulated. Now SR and CR is possible. That can be formulated. Can I look at some other route? 505B2, for example, if you have something which is given by oral route and has major challenges, a first pass effect can be given by sublingual route. So that way you are going on increasing the product line and therefore you are having product life cycle management. This way, you will also have market sustainability because your products stay in the market for longer period of time. You go on adding to the patent portfolio, and therefore you do have potential of generating revenue and profitability. As Dr. Albert Gregory said, innovation is seeing what everyone has seen and thinking what no one has thought. So everyone looks at innovations, but do you look at it differently? And if you can do that, there is a point that you can have a very good innovative idea. So obviously, innovation always starts with ideation, and that happens at university level. It has to be translated. That can be done by tech transfer offices. It can be done by university itself. You need to probably involve at this stage venture capitalists or incubators, and that is when it gets realized, that is pharmaceutically industry takes it up. So innovation ecosystem obviously has universities, and people are dependent. Last 10 years at least, we have seen lot of industries approaching us with whether we have proof of concept, and that's where our strength lies. Because in the universities you have young brain power, and you can definitely tap their gray cells, brainstorm with them, and come up with ideas which no one has thought ever. And I'll give you some case studies to understand. And this point Dr. Mishra also made, right? What you need to do is, you do have an idea and you are able to take it up to a certain level. Maybe you do pre-formulation, you do formulation studies, you do preclinical studies, meaning I'm talking about those who are into formulation, you do stability studies. But then if it has to go to the industry, that's a private sector, there's a big gap. Industry would look at whether what technology readiness level you are and whether you are able to adequately demonstrate that the product idea can be scaled up. And that's why I said, no, keep it simple. If it is simple, industry has the equipment, they'll readily take it. If it requires some capex investment, industry is reluctant. So all in all, we all have to work together. We are in academia, but unless we work along with industry, we utilize government grants, maybe utilize even NGOs at certain times, and private organization, if we shake hands, our definitely movement forward is rewarding. There are some points which I would like to tell here. We need to ponder on something. Many of the researchers do not have sound hypothesis before they start any research. Solubilization is a problem given. You will say that, okay, I'll make solid dispersions because they are easy. I'll make it up with PEG because they are easy. But whether you've done adequate literature survey to know whether your drug is incompatible with PEGs. Have you done survey to know that PEGs are autooxidative? Are there any other simpler techniques which can be taken up, which definitely will yield you results? We need to also look at, do we work in areas of unmet need? Or is it that my friends are working in this area, and therefore I work in the same area? Look at the clinical unmet needs, and the best guides for this would be clinicians. Talk to them. What are the products they are using? What are the problems they are facing? And therefore select that particular problem. Other thing is the data which we create authentic, reliable, and reproducible? One way how faculty can take care of it is give the same SOP to the next student while first student is still in the lab. If that student is able to replicate it the way first student had done, that means it is reliable and reproducible. This has to be done because many times what one person does, if you in your lab cannot reproduce, what industry will reproduce? And we as faculty have to make sure that it is a work which is of adequate standards and can be reproduced. This is for pharmacologists. Do we have good predictive preclinical models for all human diseases? Answer is no. There are many preclinical models which still need to be developed. So you might say work in psoriasis, and if you do not have good models, obviously you cannot move ahead. Pulmonary hypertension, if you do not have models, you cannot move ahead. So we need to have some preclinical models too. Also, this is for the universities or for the colleges. We should not modify rules or rewrite rules every time we wish to collaborate. They should be penned down. We should have ready drafts of confidentiality agreements. The moment industry wants to work with you, they'll ask you for CDA. They'll ask you for MOU. Do you have ready drafts to share? Or are you going to tell them that, sorry, we do not have? Maybe you can give us. That sets a wrong precedence. Can intellectual property, norms, royalty be agreed upon quickly to address translation? It shouldn't happen that every time the industry approaches, you have to think, what are those norms? How much royalty can I ask? It should be all there in your rule books. Same way, you should have written norms for milestone payments, and we should maintain log books for each instrument, equipment, and you should have provisions for data integrity. This should not be only done when there are NBA and NAC inspections. This has to be done throughout the year 24/7. And all our instruments have to be CFR compliant, so that is when industry will come to you. If you do not have 21 CFR compliant instrument, why will they rely on the data? In fact, even for characterization, many industries approach us only because we have CFR compliant instruments. So in order to save money, many academicians will not go for compliance, but it is a must if you're looking at industry coming to you. Other things which need to be considered, do we have GMP setups? None of the academic institutes in India has GMP setups. In that case, can you identify quickly where can I do a pilot plant scale up? If they ask you to do, for example, many times we give them when we are ready, at 1 kg scale, but they want us to demonstrate at least 5 kgs, 15 kgs. So you need to have GMP setups. You need to ensure confidentiality from students. So I sign CDA with students also, because they are the ones which will be employed by industry. What if they share your trade secrets? Take adequate survey from end user. This helps you make a product better. For example, if you're working in aerosols, no, mothers are the right people who will tell you what are the problems they are facing and how can you modify the device for their kids. And of course, we all academicians have to dissolve our ego. If we need to work with industry, please dissolve our ego. This doesn't mean that lower your rates. No, I'm not saying that. I'm saying be a listener also. Typically, professors feel that they are one up, industry does not know anything. Industry is aware of rules and regulations. You are aware of your technology, so shake hands. And of course, take clinicians' perspectives. So these are the expectations, I would say, from the industry end and the university end. University, when they're shaking hands with the industry, should ask for equal partnership, which never happens. And in fact, you need to ask, then only they'll give you, right, the amount which you are asking for. Not only that, you can ask industry whether they'll be able to place our students there. Take up, tell them in advance, which goals which they are expecting are feasible at our level and which are not. It is okay to tell them in advance that this is what I can do and this is what I cannot do. They will appreciate that rather than you taking and then, going all around India to see whether I can do it or not. And of course, industry gives you enough funding and infrastructure. But on the other hand, industry always wants data integrity. They are looking for quick solutions. Most of the industry will come, most of them have come to me when they work for it on a project for a year and they are failing. And then they say, we have only 6 months now. So they are looking for quick solution. So 6 months with 3 months stability data, meaning on your hand, you have only 3 months. They are ready to give you chemicals, consumables, everything. But idea has to be yours and you have to move quickly. Of course, they are looking for skilled manpower and always they look at market perspective. So these are some things which I wanted to tell you, before I tell how we go about in VBP research group, what are our interests. So we work in varied areas because I get students who have biotechnology as their background and I do get students who have technology as their background too, besides BPharm. So as a result, we work in diverse areas, but yeah, and we've been benefited by working in diverse areas. So this is one case study which I'll take of brucellosis. Brucellosis is caused by B. abortus, and this particular disease affects not only animals but also human beings. Animals are the source of infection to humans. So more than 5 lakh new cases of brucellosis are reported worldwide each year. Now, what did we do? We understood the disease condition. We knew that it primarily affects cattle, and the side effect is abortion, which is bad for those who are catering to animals. And most of the time, diagnosis in animals is delayed. If it is delayed, it affects other animals, and it also affects the farmers who are working with them, the doctors who are working with them, and those who take milk products and dairy as well as the meat products of these animals. So it's a big problem. There are certain diagnostic techniques like agglutination, complement fixation, rose Bengal, all these tests are there, but they give you non-specific results. So what gives you specificity is PCR or ELISA. So just like COVID, PCR or ELISA is being done using animals' blood sample. So here we understood this gravity, and what we came up with is point of care diagnostic kit. Now this point of care diagnostic kit is very, very simple kit which can be assembled within the lab. So you do have, of course, if you see this downmost figure, you have a conjugate pad onto which you are having 2 control lines. On one end, you put absorbent pad so that any excess can move upfront. What is there on these lines? One line is a test line and one is a control line. As you can see on the control line, you do have antigenic fluorescent silica nanosensors. And you do have anti-lipopolysaccharide IgGs. And then here, on another line, you again have these FSNs, but you have anti-Brucella IgGs. Very simple, just work
Fostering
like
research and
pregnancy
innovation in
kit,
pharmacy, advancing academics and
wherein you need to apply your sample here. Now this sample, what is that, what we have done? People were using blood. We said we'll use milk of animals, or we'll use urine of animals, or even saliva. Anything can be used. So it becomes now point of care. That means even farmers can do it at their facility. So you apply the sample here, the sample will move forward depending on whether it is positive for Brucella or whether it is negative for Brucella, you will get the results. Obviously, if it is positive, you'll see that both the control and test line will fluoresce. And if it is negative, only one line will fluoresce. So this patent is now granted. What is it? What we have? We have fluorescent silica nanosensors. Now, why silica? People are using gold, right? Now, gold is expensive nanosensor. We thought, can we get silica? Silica can be, from bagasse also, sugarcane waste also. We made silica, made it fluorescent, and use it for ICDT. What are the results we got? Obviously, we first confirmed that LPS gets absorbed onto FSN surface. This is the results on humans. We've compared it with all other techniques. You can see that ICDT, which is standing for ICT diagnostic test, or also you can say immunochromatographic diagnostic test. It is sensitive, it is specific, and it is in agreement with PCR and ELISA. It's done in urine as well as in saliva samples. We did it on around 103 cattle and 30 buffaloes. And there also you can see ICDT compares very well with ELISA and PCR. A simple test which compares to ELISA and PCR. So what is the novelty? We have point of care diagnostic test. We have used silica nanoparticles, which are cheap. It is a non-invasive method. You don't have to take blood of samples. You can take saliva, you can take urine, don't have to, meaning be it humans or be it cattle, you can rely on their non-invasive technology. And for the first time, we have used fluorescent silica nanosensors on lateral flow analysis format. You need to check whether it has a competitive landscape, if you have to go to market. So currently, in fact, studies in New Zealand have been completed. The technology has been transferred already. What I have not shown you is how to make silica nanosensors. But what I have shown you is that how the hypothesis was created, right? So this particular landscape, if you see, ELISA kit is being sold per kit $527 to $715. And what is the cost of our kit? Rupees 20 only. And that is what is going to give competitive advantage to all those who would be using this kit. We have not given exclusive license to anyone. It is a non-exclusive license, meaning if 3 manufacturers want to take it, to all 3, I should be able to give it. Advantages, of course, to university and to us also. So in this slide, I have just shown how our kit is definitely taking care of accuracy. No advanced facilities needed, no blood samples needed. It is not time consuming, and of course, not expensive also. Moving in the same direction, what we have also done is made a nano vaccine for this because we understood that's a grave problem. Currently, even to the pregnant women, I'm talking about humans, pregnant women, what is given are antibiotics like rifampicin, which is not correct. And of course, to veterinary, cattle, etc., they are to be slaughtered. So what we did is we thought we'll make vaccine under One Health approach. One Health means that something should be useful to animals and to human beings. And this particular vaccine, so that it doesn't require cold chain storage, it is easy to produce, we made a nasal vaccine. And what is that? It's a subunit vaccine for brucellosis, which comprises submicron size mucoadhesive, biodegradable polymer based nanoparticles, and what we have used is a green technology. Very, very simple technique we have used, and that technique is, in fact, this paper has also been published. What we have done is we have used REST technology, rapid expansion of supercritical solvent. We take everything in rest chamber, pass supercritical carbon dioxide through it, and it passes through atomizer to give you nanoparticles. One step reaction. And that is why this was appreciated even by Department of Biotechnology, and we use the same technology for nasal COVID vaccine too. Simple technology, very, very highly scalable. Particle size and nano range and reproducible, very reproducible, patent already granted. So the results of this, definitely you can look at that. This graph indicates the size is in nano. It is mucoadhesive, stable. In fact, even for 1 year room temperature stability, we have completed. We've looked at in vivo immunogenicity. For this, you don't even just look at serum levels. No, serum IgG levels need to be checked. But also what happens, the nasal fluid needs to be looked at because we are saying that it would be administered by nasal route. So we have got prolonged immune response using this particular technology. Having done that, we had to do protection study also. That means whether really, if you give a challenge, brucelli organism is given, whether animals would be protected or not. And that study results also are very highly encouraging. And if you look at IgG1 and IG2 titers, anti-LPS titers in mouth serum, nanovaccine group stands out. Now, what is it? One is that, yes, we have used one pot technology. Secondly, we have used, natural adjuvants. And those adjuvants, natural polymers, natural adjuvants, everything available in India. So it's an indigenous technology which can be used for many vaccines. This slide just gives what is the log10 IgA and IgG1 titers. And you can see that definitely statistically significant levels were detected in groups 4 and 5. And 4 and 5 are the ones which are with our vaccine. Bacterial burden analysis also indicated that vaccination protects against splenomegaly, which is a major problem with this particular condition. So this way we have concluded that we do have a green vaccine with reduced bacterial tissue burdens, reduced inflammation, increased anti-LPS titers. Next study which I'm presenting is for malaria, wherein we have used a combination used orally, and for the first time in world we've made a IV formulation. The technology already has been transferred again to industry. So if you look at WHO website, artemether lumefantrine is given as a first line strategy. It's a recommended therapy, right? But problem is if people have cerebral malaria, there's no time for oral absorption, so it has to be given by IV route. But drugs doses are such and their solubility problems are such that no one thought of making a combination IV formulation. And people die from 100% mortality generally seen from severe malaria. So we made nanostructured lipid carriers and looked at its in vitro and in vivo efficacy. You can see that our formulation has protected as against the controls where malaria parasites grew, as against a marketed formulation where, as far as treatment was given, it was under control. You withdraw the treatment, counts again rise. But in our case, it gave us 0% parasitemia, which indicates that this formulation is very effective. We also use another protocol wherein, kindly mute yourself. We first generated malaria in animals and then started with the giving administration. And in this case also, our formulation performed better. Now was a time to move to cerebral malaria, and this is done in C57 mice. These are not normal bulb C, right? Not Swiss mice, it is in C57. Here again, we could see protection. This patent also is granted, and patents, a very broad patent we have taken on nano drug delivery for treating parasite infection. So it covers all the parasites, not only malaria. Proof of concept is seeing it. You can see here RBCs which have granular mass are the ones which have parasite in it, and the plain ones are which are normal RBCs. This red thing is our NLCs which are actually having dye in it. When I start the video, you'll see that they selectively go wherever there is a granular mass. So they get localized specifically only where there's a granular mass. They leave the normal ones aside. So we've done a lot of study to prove the mechanism of it, and these studies have been done with TIFR. If you want still better, proof, look at the marketed group. Marketed artesunate is an unstable formulation, and after treatment also, animals are paralyzed. You can see how they are walking. And this is our formulation. After giving the formulation by IV route, they recover. In fact, the veterinary scientists told us that can be used for another study. They are so very normal. So this is a proof of concept. The formulation has been scaled up. Industry has been shown all the scale up, and definitely, the clinical trials are going on at their end. Some simple examples before I end. People will say, is it always that nano has to be done? No. One product which we developed for equine scoring, horses. This was done with Ireland. And that also during COVID period, we did brainstorming. Luckily, I could identify my own student who was doing, B.Pharm student went to do masters in Ireland. He just completed. So every night discussions and every day he used to go and work in the factory premises. In 3 months, he scaled up the product to 5,000 liters. Now, what was the problem? Equines, they die within few hours if they are untreated, if they have diarrhea. It affects more than 20% foals within first 6 months of birth. Typically, antibiotics are given to these young horses, right? Foals is young one. So definitely there was a problem. So we identified, the company came to us whether we could come up with something. And if you remember, in our dispensing, we used to talk about kaolin-based suspensions. We gave them a probiotic-based suspension, which can be used to treat severe dehydration, can take care of acid-base balance, and of course, the natural gut microbiota also it can restore. So very simple solution after identifying the unmet need was given, and the product, this particular product also is now being sold in more than 10 countries. by different trade names. This is one. Tufol is just one. So this is nothing but a unique blend of clays, prebiotic, and electrolytes as a stable oral suspension. Same suspension which used to do in mortar and pestle, I would say, we have added a prebiotic and of course done it in such a way that's a very stable suspension. Their eyes, were open. They did not know how to formulate suspension. Our suspension was so smooth, which they had never seen. So this as is given as a complementary feed so that foals maintain their gut motility and therefore they are benefited. As I've already told you, it is a non-antibiotic based formulation. Another one which we did under Startup India collaboration, the last one, the dental alginates. Now, dental alginates were being imported. There's a global market which is huge, but still imported. So this company who had got, grant for making dental burr. Burr is a device which cleans up your tooth, but then he had money, thought what could be done, and came to us. PCM Health Sciences, they came to us. We collaborated and we made a hydrocolloid system. Now, alginates were marketed. So, what is it? What different we did? No, from the market. These are the marketed packs of our developed product. Marketed ones were available in jars, 1 kg jars. We changed the packing and we also gave unit, the way we have unit dose systems, we gave these small packs. From the major pack, 1 kg pack, if you take one scoopful, this has to be mixed with water, and dentist has to keep looking at the time before putting it in your oral cavity to take dental impression. And then again look at time, see whether it is dried and take it up. What we did? We added 2 indicators so that dentist doesn't have to look at time. When the indicator changes color, he knows it's time to put in the dental cavity. On the teeth, it will change color on its own once it dries, and then you can remove it off. Not only that, kids used to throw it off, obviously, because of the taste. So we gave many flavors, but 2 flavors which are doing very well in market are strawberry flavored and chocolate flavored. So simple things with a difference. It's not that you always have to do something which is very, very novel, but it can be simple, incremental innovation which can benefit India. And this is the last one where we have made a stent. Stent is something which is implanted in the artery so that it can open up the artery. Those people who have plaque, they are prone to angina pain. If it opens up, the blood can easily flow. I do not have much time, but what I'll do is show you the studies here. Animal studies data indicated, these studies have to be done in pig. It indicated that formulation was very effective because the arteries remained open. They did not close back. Then the study was done in humans. In humans, it again indicated that drug-eluting stent, you can see, had very, very low incidences as compared to bare metallic stents. And this is the proof of concept. When we did study compared with Abbott stent, the arteries remained opened. And in fact, a 3-year follow-up shows that our stents are much better. So never believe that India can't do anything. What was the difference here? What was the USP of VBP group, and which obviously worked with Sahajanand Medical Technologies to give the technology to them? Everyone was using bioerodible polymers as coating. We used biodegradable polymers. Bioerodible, if they erode, they can form loci for thrombi formation, because of which again there'll be heart attacks. We thought if slowly it biodegrades and becomes part of your body, obviously that would be much preferred, and that became our USP. The second USP was we gave one final coat onto the stent, which gave it so much good lubricity that clinicians or the interventionists, who passed the stent did not have any problem passing it through any of the arteries. So that became our problem. So these are some of our products which are already in market. You can see n number of products, the stent products which are in market. We have given IUD, that's intrauterine device, also to basically a company. We gave it to a company, but they gave it to WHO. And then there are many products which are nano-based, non-nano based, which have reached market, as your orator said, about 20 plus technologies. And all this would not have been possible if it was not for collaboration, grants from government authorities, grants from private authorities, and collaboration with many companies, be it in India or abroad. This is our VBP research lab, has everything, whatever is required for formulations. And of course, all of us work with this motto. In a nutshell, it means that focus. Once a problem is given to you, focus only on that. Leave everything else aside. So, with this, I would like to conclude my talk saying, in words of Albert Einstein again, that imagination is the highest form of research. Imagine, come up with a technology which no one has thought, and you will definitely do wonders. Thank you so much once again.
Thank you very much, ma'am, for sharing your research insights about brucellosis, chromalgin, malaria, quine, and many more products. Lot to come more. I think you were short of time. And showcasing the translational research and guiding on us how to collaborate with the industries and bring something to the society that is helpful to the people. And your advice on selecting meaningful research experiences, it is truly inspiring. And we are deeply appreciate you for the time and effort you have spared for us. And this is, I think, the most important line which I want to say.
Thank you.
Because sharing experience with you all, we get something new every time. And it is very helpful to us.
Thank you so much.
Every time whenever I listen to the persons like you, I take at least one thing from it so that I can go for it. Thank you very much, ma'am. Thank you very much.
Thanks, Dr. Priyanka. Thanks once again to Guru
The
Gobind.
background.
Thank you so much, ma'am.
Thank you.
Yes.
My sharing has stopped, right?
Yes, ma'am.
Priyanka, go ahead, please.
Yes.
Ask it.
Now, I would like to introduce our second speaker of the day, that is Dr. O.P. Katare, who is Professor, University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. He is globally recognized academician and scientist with vast experience in teaching, research, university administration, pharma industry, and guiding youth for the excellence and leadership skills. In his 3 decades of experience, he has contributed immensely, which is reflected in terms of number of quality publications, including research papers and reviews, edited books, Indian and international patents, technology development and transfers, commercialization of novel pharma products based on nano and liposomal technologies, and multiple government and industry-sponsored research projects, bringing of worth crores of rupees, bringing lots of financial and infrastructural resources to his organization. And besides this, he has many other recognitions to his credit by national and international agencies like OPPI Best Scientist, IMB Best Patent, IES Bhopal National Teacher Excellence, DVS Jan Punjab University Best Scientist, Faculty Research, MHRD, lifetime achievement and illustrations alumni of Dr. H.S. Gaur Vishwavidyalaya APTI Best Teacher Scientist Awards. Professor Katare, in true sense, is the aspirant of serving the society with utmost humanity while chasing the highest spiritual goal that a human life so offers for everyone. I welcome you, sir, and request you, sir, to please take over.
Thank you, Dr. Geeta. Am I audible, Dr.
Yes,
Gaurav?
sir. Yes.
It's a great pleasure be with virtually at their Yamuna Nagar. I visited Yamuna Nagar campus probably in the beginning of this college for the appointment purpose. So I had a very uh, feeble memory. So the topic which I chose, looking to the aspirations on campus, led by Dr. Gaurav and team. And I said very bright faces. I heard a lot about Dr. Gaurav. We have the BN College of Pharmacy. So the uh, where I started my journey, academic journey. So it all matters is aspirations that brought me here at Punjab University campus that started at Udaipur BN College. I was also serving like uh, any one of you, some private campuses, but aspirations brought that uh, what is my destiny. So uh, what I mean is that a campus which is aspiring, the people which are aspiring there, it's a perfect ground to talk about the topic. Academia, the breeding ground for research and innovation. How to build it is a $1 trillion question. सभी लोग कहते हैं कि अभी आज की स्टोरी ये है, टॉप स्टोरी यही है कि if research and innovations to come out, they will come out like US and other foreign countries from the academia. In foreign countries, we see already that the universities are the churning ground. They are the churning the machinery for the innovations, new molecules. But in India, somehow in India, I mean in our Bharat Mahan, we are lacking this. मतलब हम उतने अभी महान नहीं बन पाए जितना हमें होना चाहिए था. तो ये एक बहुत सेंसिटिव मैटर है कि हमारे पास अब इतने हजारों हजार कॉलेज हो चुके हैं. अब उनको हमें कैसे हम उनको ट्रांसफॉर्म करें, कैसे हम उसको कन्वर्ट करें. सो कहने के लिए तो आसान है कि एकेडमिया में ऐसा होना चाहिए, वैसा होना चाहिए, बट एक्चुअल में कैसे होगा और उसके लिए क्या मतलब करना पड़ेगा, ये बहुत इंपॉर्टेंट एस्पेक्ट है. लैक तो हम बहुत जगह कर रहे हैं, बट वी हैव टू आइडेंटिफाई दोज डायमेंशन. समटाइम्स दे आर मिसिंग, दे आर हिडन, दे आर कॉल्ड एक्स फैक्टर्स. पता ही नहीं होता कि क्यों नहीं हो रहा है भाई, पता ही नहीं लगता है. और हर किसी को अपना कैंपस, अपना इंस्टीट्यूट ऐसा लगता है कि हमारे अंदर ही कमी है और पता नहीं चल रहा है कि क्या क्या कमी है. अरे हमारे यहाँ का तो माहौल ही ठीक नहीं है, इकोसिस्टम ठीक नहीं है, लोग ठीक नहीं है. ये बहुत बड़ी प्रॉब्लम है. तो एक्सीलेंस आए तो आए कैसे? नॉट ओनली फॉर रिसर्च एंड इनोवेशन, it is a substratum, the ground, the total mahal, the ecosystem, that it matters. So I brought this story for, so what I am going to talk first, something which I have done personally, not like Madam Bandhana Patrawale, because she is the storehouse of those tech transfers sitting in Mumbai, the pharma hub of the country. It's difficult to match Vandana ji. But with our humble efforts at Punjab University, Chandigarh, we could do something. समथिंग ना कि थोड़ा सा शुरुआत कहीं से की है हमने, एक स्पार्क जैसा हम लेके आए हैं। उसके पीछे क्या कहानी है वो मैं स्टोरी आपको सुनाऊंगा, और उसमें फिर उसी से हमारे कुछ चीजें निकलेंगी, अपने आप ही निकलेंगी कि हमें करना क्या चाहिए। तो मुझे भी ज्यादा पता नहीं मैं क्या बात करूंगा, क्या बोलूंगा, बट इट्स एन अटेम्प्ट कि हम देखना चाहते हैं कि वी शुड आल्सो बिकम लाइक आईसीटी एंड अदर leading institutions worldwide. So Punjab University, what you see in front is a campus where we salute, we worship the spirit of Mahatma Gandhi. This is a Gandhi Bhavan, the beautiful architect that designed by De La Corbusier, a French architect. And those times, the gift to the country, Chandigarh, and the gift to the Punjab University is this beautiful monument. So, saluting this, I move forward with the slide. हाँ, ओके। तो सबसे पहला तो हमारे जो जहन में बात है, एटीट्यूड। एटीट्यूड मैटर्स मोस्ट। हमारे दिल में एक तमन्ना होना चाहिए कि भाई मुझे, और एक not only for research and innovation, we should be खोजी by our attitude. We should keep asking the questions, the quest, until unless that is there, we cannot do anything. सबसे पहले हमारा एटीट्यूड है खोजीपन का होना चाहिए। खोजी होने का मतलब क्या है ये हम आगे देखेंगे। It is only, it is you see who is खोजी। जब खोजी की बात करते हैं तो न्यूटन का नाम सामने आता है। अरे man, all were seeing that apple falling down, but this man only who was who had this question, why it is. Otherwise, all were saying कि ये तो गिरता है और गिरना क्या है, इसमें कौन सी खास बात है। हम लोग भी ऐसे ही हर चीज को एक्सेप्ट कर लेते हैं, कभी नहीं सोचते उसके पीछे कारण क्या है। वी एक्सेप्ट एवरीथिंग इन सराउंडिंग कि ये चलो ठीक है। अगर हमारा ऑब्जर्वेशन है, द पावर ऑफ ऑब्जर्वेशन एंड द माइंड, वेदर न्यूटन, आर्किमिडीज, कैकुले, what is common to all? The common is the relaxed state of mind. Even Kekule was not even relaxed, he was sleeping to get this idea. Can you wonder what he was searching? So the innovation comes down, the research comes down sometimes. We try to upload, but sometimes it is a downloading of the information in the relaxed mind. So in the beginning, I introduced this concept of mind, or the role of the mind, which is very, very critical in making your campus, each and every entity according to that, to build that ecosystem. So the gravity man से हमने ये सीखा कि हमें क्वेश्चंस पूछना आना चाहिए और बहुत गहराई से सोचना चाहिए। सो लिटिल स्टोरी, थोड़ी सी कहानी है हमारी। दैट इज in front you, what you see is the department, University Institute of Pharmaceutical Sciences, which is known for our father figure, Padma Shri, late Professor Harkishan Singh. the only molecule given to the country by any institute, university, is this chandonium iodide, for which he got this he was honored for the Civil Award, Padma Shri. So our department salute, and this is the kind of a spirit that it prevails and nucleate the atmosphere, the ecosystem. So in this atmosphere, I joined Punjab University long back. It is down the lane almost 27 years. So I was just wondering whether I can do something which is worthwhile to be useful for society. Not only your academic research, but the research should turn into innovation. Innovation which is used by the society. सोसाइटी के कुछ काम में भी आए. ऐसा नहीं है कि हम सिर्फ पेपर पब्लिकेशन, इम्पैक्ट फैक्टर, ये सभी बात करते रहे. तो दिल में एक तमन्ना थी, तो हमने इसको ज्वाइन किया पंजाब यूनिवर्सिटी in 1997. And I was coming from the very humble background. This Punjab University is on top, and I was coming from Gandhinagar, Gujarat. That time I was serving Gandhinagar KB Institute, and before that I was in BN College. So there was a lot of apprehension, lot of things कि क्या करेंगे, कैसे होगा, और कर भी पाएंगे, whether I will be able to sit along with those high profile Professor Kulkarni, Professor Handa, the luminaries of the pharma world. बट दिल में एक तमन्ना थी और चाहत थी, एस्पिरेशंस थे, खोजी का एक एटीट्यूड था, तो वो मैंने काम शुरू किया. सो what did I do? Research, because this research was on mind. Besides academics, we did a lot of thinking कि क्या करें. तो गांधी जी की एक तस्वीर, जो पिक्चर मेरे सामने आई, वो आपको शेयर कर रहा हूँ. कि मोहनदास करमचंद गांधी जी जो अपने थे, उनने ये बोला कि आई वुड लाइक टू डाई बट विल नॉट टेक केमिकल्स एज मेडिसिन्स. मतलब वो मरने के लिए तैयार थे, लेकिन कोई अंग्रेजी दवाई खाने के लिए तैयार नहीं थे. ये बड़ी अजीब बात थी, लेकिन उनको पता था कि ये केमिकल्स क्या कर सकते हैं बॉडी में. सो गांधी जी, Bapu Gandhiji is still stand tall before us with the clear-cut statement that the drugs are not safe, they are not effective, and they are actually lacking compliance. तो ये issues जब उनने कहे थे, अभी भी हमारे सामने हैं, and we are struggling. We see that we suffer sometimes from drugs more than diseases. तो डिसीज से ज्यादा कभी ड्रग्स भारी पड़ जाते हैं, अभी भी हम देख रहे हैं. सो गांधी जी गेव अस द चैलेंज. Challenge means the opportunity for us. Actually, we cannot simply accept, because we are in this discipline, and it is our dharma to explore and to come out with the solutions. जो भी प्रॉब्लम्स हैं उनके लिए हमें सॉल्यूशंस लेके आना है. तो what are the solutions? I would like to, now coming from the pharmaceutics, drug delivery, formulations, this is the field which belonged to me. So I started working for the drug delivery. Drug delivery, all must be aware that current drug molecules, they are just dumped. They get jumped, they get bumped, and they get dumped. कहीं डालो कहीं जाते हैं, वो कहीं पर भी गिर जाते हैं. तो इससे सारी समस्याएं होती हैं. So we need the right course of their kinetics, the distribution, the dynamics, the targeting. They should have a right trajectory. They should have right trajectory and the trajectory that the molecule should be delivered. So the Paul Ehrlich, the famous Paul Ehrlich that drugs can be magic bullets, but herein we could not find that magic bullets, but we can have them by magic guns. Magic guns means delivery system. तो अगर हमारे पास में व्हीकल है, टारगेट कैरियर्स हैं, तो हम उनको कोशिश कर सकते हैं. So this is how the new lease of life is given to the old drugs, amphotericin B, and many examples are now there where formulation science, so takes drug delivery, nanotechnology, interdisciplinary it has become. Engineers have come and now AI is there. So now we have a lot of hopes that we will be able to hit upon the target, particularly in case of those diseases like where cytotoxic drugs are used, cancer and all, and otherwise also in any. So we need the right course of the delivery. So we have liposomes. You all know about the liposomes by now because it is introduced even in undergraduate. It's a vesicular system where it has the range of physicochemical this acceptance when it can host because of its unique range of the values, the HLV values, the various solubilities, the various it can join, it can give the accommodation to wide variety of molecules. So the technology, which is very where I graduated through all my this PhD program, and probably I was the first one to start the PhD in this liposomes. Mine was the first thesis at Sagar University, thanks to my guide mentors, Professor Dikshit and Professor Vyas. And little bit of the technology, I will not talk much, but all that it matters is the building blocks, the phospholipids. The phospholipids, they are the main the constituents. They give rise to the kind of interactions. So that turns out to be supramolecular self aggregates of these association collides. So because of the two chains and one polar head, they provide the unique tubular structure which come together to form this membranous structure, and because of this they turn out to be the having lot of unique merits, their entrapment diversity, solubilization capabilities, potential for delivering and targeting, toxicity, safety, compliance, बहुत सारे इसमें खूबियां हैं. And so they're the one which can be explored and exploited in many ways. The diversity is there. You just change the composition. You just change the process, the various factors which influence their making. The same material, the same formula, the same composition can turn out to be a very different structure by their in their making. So without changing the same formula, they can give you the diverse range of products. That is the beauty, and that provides the novelty, that provides the scope for the inventiveness and for the patents and for the this special applications. So unlike the other conventional dosage forms and delivery systems, they have multiple the factors, the multiple ways of their standardization. phase transition phenomena. That's a unique thing in it. And because of that, we have the possibility of targeting using pH and temperature and other energy stimulus. So they can be modified on surface and so on and so forth. And this is how they are, the technology, the liposomes are characterized, standardized. I'm not going into much details. There are high-end techniques like TEM, and they are used to characterize, and HPLC and all that for analysis purpose. So there are products in the market. That give us the confidence that yes, something is done. Otherwise, people kept wondering कि ये चीज हो भी सकती है कि नहीं हो सकती है. When I started working, I am to share. It's a pleasure sharing that people were not believing that such structures can exist. And once I was working on bench, the professor from South, Professor Rambabu, visited our lab, and he asked my guide, Professor Vyas, कि ये सही में बनते हैं क्या? So he took me to he took him to my table, and I could I could show him that under the microscope these structures are there. Not only they are there, but they can capture the microbes when they were mixed with the slide. So it was it was that time that people were not believing. I mean, even when I started working here at Punjab University, people were commenting like कि वो तो बनते ही नहीं है, फट जाते हैं, उसका तो कुछ हो नहीं सकता है, it is futile to work. And but challenges are there. अगर आपका attitude है, आपका तमन्ना है, तो आप कर लेते हैं. So I started working on this कुछ एक प्रोजेक्ट कैसे शुरू किया जाता है, and how to choose. That's a little bit of it. So I selected one drug, the ethanol, and the disease, the psoriasis, and the dosage form, the technology I chose, the liposomes. So psoriasis is the disease which is very, very difficult to treat. The wide population, बहुत सारी population 2 to 3%, 130 million, and we have 75 associations. Even one association is there for all associations, the confederation of these associations. So we have these drugs. Mostly the drugs are given orally or by injection. They destroy, they actually create problems all over the biology inside. So and topical therapy is not very effective. So there is a medical gap, and a medical need is there. So before selecting any disease, we have to see whether the project demands or not. So it befits into that. So we started working, identifying this molecule, and this is the molecule which was banned twice in past in the history because of its non-compliance, its toxicity or safety, and people did a lot, 2,000 papers, several patents, exclusive meetings, and but they could not find the answer, and it was banned twice, but the doctor brought it back because there was no substitute. Though it is non-compliant, irritating, and staining, but it is actually very, very effective. So because its efficacy is weighed down by toxicity. So its efficacy is because of the multiple ubiquitous targeting all over the places, cellular, intracellular, बहुत सारे इसमें टारगेट्स अवेलेबल हैं और ये सभी जगह हिट करता है, इसलिए बहुत अच्छी ड्रग है. बट the same time it is fearful drug free radical formation. So the idea came to me through PGI. This is how it's very, very important. The partner, the medical partner, the doctor. There's a there was when I started here the work, the head of the dermatology, this PGI, Professor Bhushan, I was visiting. He gave the idea that why don't you work for this product, which is really very good US vitamin product, diethanol, but it is non-compliant. So he gave the idea. It came from the medical side. So this is very important for research and innovation. We must gather, we must seek, we must come to the interface of the doctors. कि करना क्या है? So this product, I started working and found that there is an interesting note from the most जिसने सबसे ज्यादा काम किया है मुस्ताक एलियो के who wrote in 1992 in the journal that it is doubtful whether an entirely non-irritating diethanol derivative or treatment modality will ever be developed. मतलब ये भरोसा ही नहीं था because they need some irritancy in it. So because of this, they in desperation he wrote that it is not possible to improve this molecule. Actually, what the people were doing, the researchers were looking at is the chemistry. Normally, when there is any problem in the molecule, we always try to temper with the functionality. Their group इधर का लगा देंगे, उधर कर देंगे. Chemistry, with all due regards to our chemistry friends, I have a lot of respect for the chemistry because without chemistry we cannot move forward. All our physics depends on chemistry and physical chemistry. But here is the case where it probably chemistry did not work. Why? Because the moment you tamper with the molecule, you get the less of this side effect, irritancy and staining, but at the same time their functionality is also lost. So efficacy also goes down. So it is just like that. The molecule needs some sort of aggression, like a wrestler. When a wrestler has to fight, he has to have some aggression, anger. So this angry molecule, when it loses its loss, its anger, I mean functionality. So efficacy is also lost. You can see that in the exoline, this happened. So chemistry will not work here, and Sutics approaches which are conventional, the vehicle modification and other clinical modalities, they also did not work. Probably it needed, they improved the evolved form of pharmaceutics, the drug delivery based pharmaceutics. So we started working, and the first time we found that what is the problem? Diethanol. It was never reported earlier. So it is what is the paradox? Paradox is that it has the reactivity, super reactivity, which it needs for the action, but at the same time, it is harmful. So ketoenol tautomerism, that it goes to the first generation, second generation, and third generation. That is a very quick degradation of the molecule. So we started thinking over it, how to make it, and when so what is there which comes in the academia, in the academic room, classroom situation, it is an example how this academia can be a breeding ground, because teaching is very important. It is only in the classroom I got this idea. Why not to use these phospholipids, which are there in the lecithin, to be used for the molecule to moderate its reactivity. So probably I was not looking for the drug delivery, but the moderation of the drug reactivity. It's a different thinking. In those days, there was no one who were working on topical, and there was very little idea that even topical will need some drug delivery concept. So sometimes people will say कि ये क्या है, topical पे काम कर रहे हो, इसमें तो कुछ क्या है करना है. But believe me, it is very difficult to make it. And there's a topical sometimes becomes so heavy because of the many problems we face across. So I was in the classroom and teaching physical pharmaceutics, and the idea descended into my brain while I was struggling to teach this antioxidant properties of the lecithin, the phospholipids. So we did it, and we found that the systems, the real time, this picture of liposomal entrapment after diethanol, one of my PhD scholar, the first PhD scholar from Udaipur BN College Pharmacy, whom I taught, Dr. Ravindra Agrawal. He joined me for PhD program, and he started working. He really did very hard work, and his aspirations were really very appreciative. He did a lot of work and collaborated with the PGI, and so it did it. से काम हो गया, and we got this design, the unique designs to fetch us the patent worldwide. The company came forward to support us for the patent and all. In those days, there were very little were actually in the practice about this patenting. बहुत कम ही लोग करते थे. पंजाब यूनिवर्सिटी में हमने पेटेंट पहला किया था, तो there was a lot of noise, है ना, and so now there is a flow of patents. ना, अब तो इतने patents होते हैं कि people there's like a river of patents flowing. But my question only 2 days back, when we had a brainstorming session, I had a question, and people will ask questions worldwide कि आपके इतने पेटेंट हो गए हैं, उसका हुआ क्या? पेटेंट अकेले कराने से क्या होगा? And patent is a commercial document. Actually, it's for the commercial value. So until unless it comes to commerce, what is the good use of patenting? It's a wastage of resources. So patent, अभी हम देख रहे हैं कि बहुत से पेटेंट ऐसे भी हो जाते हैं जिनको कभी भी वो नहीं हो सकते हैं प्रोडक्ट में कन्वर्ट. स्टेबल ही नहीं है तो कैसे कन्वर्ट होंगे? तो वो स्केल अप ही नहीं हो सकते तो कैसे होंगे? उनका मटेरियल ही नहीं है तो कैसे होंगे? तो ये सारे डायमेंशंस है ही नहीं उसमें कभी. ये इनकी अकाउंटेबिलिटी तो है ही नहीं, उसकी नजर ही नहीं है. दे बिकम ऑल एक्स फैक्टर्स. फिर होता है कि यार इतने पेटेंट हैं, वो तो एक्सपायर हो गए, और उनको अब रखने के लिए भी पैसे लगते हैं. So what I was actually talking about this, how did it happen that these liposomes could turn the table around, or that it reversed the trend of degradation. It slowed down actually. So the actually required anthracycline radicals were more than that of the toxic and side effect radicals. So we had this TGI conducted clinical trial. So thanks to this academia collaborations with the Punjab University PGI collaborations, without spending even a single penny, we got 21 patients studied, and we had a wonderful results in a reduced dose vis-a-vis US vitamin. So thanks to the Derma PGI, that gave us this wonderful beginning of my career here at Punjab University. So we had a one company then. So we didn't go to the industry yet. Still, we are not going to the industry. Industry is coming to us because of their the proof of the concept at the clinical level. Industry comes running, and otherwise that's very difficult. So industry academia collaborations, people talk a lot about the interface का एक दूसरे के पास आना चाहिए, ये करना चाहिए, वो करना चाहिए, लेकिन जब ही आएंगे वो जब आपके हाथ में कुछ worthwhile होगा, when you have something worthy and some value in your hand. So thanks to this, we had a company, we got the tech transfer. शायद ये पंजाब यूनिवर्सिटी में इस तरीके का पहला ही टेक ट्रांसफर था, इसके पहले भी हुआ था हमारे डिपार्टमेंट में. So Professor late Professor Singla got it, and thank God to Panisha Tech, and that was actually a role modeling story that inspired us. So, but that was a technology based, nanotechnology based, the liposome technology based. This was the first one. We got the paper signed, tech transfer, and we got the scale up. तो लैब में बनाना आसान है, लेकिन जो स्केल अप के लिए हम जब जाते हैं ना, तो रियली इट्स बिकम्स द ह्यूज चैलेंज. उसमें बहुत सारी डिफिकल्टीज आती हैं, एंड वी रियली नीड द टीम वर्क, कोऑपरेशन, कोलैबोरेशंस, म्यूचुअल ट्रस्ट और कोहेजन. ये बहुत इम्पोर्टेंट है. अगर इसके बिना नहीं हो पाता. यहाँ पर हमें नॉट ओनली टेक्निकल, नॉन टेक्निकल सपोर्ट बहुत ज़्यादा चाहिए होता है. तो टेक्निकल एंड नॉन टेक्निकल, इट मीन्स इट्स ए टोटल ecosystem that it needs. Otherwise, it's very difficult. There are many stories, which failure stories also, which I faced for scale up especially. So we had this, the product came to reality on the name of Sorizome. So this is the first product marketed, and but we could not capture that market because of the lack of market muscles of the company. But it is still a story, still a gold standard story, which it has maintained its glory, and the Sorizome was launched, and it is available in the market. It is available, but it is not. It could not pick up the market. No matter, I did not lose my heart, and kept on working for the further on my further aspirations with the help of the students and our colleagues and the ecosystem which we have created. And we got the next with the same company, this Koltar product, where this time we didn't go for the liposomes. We went for the complex nano systems, which made up these of course, by phospholipids in combination with other lipids, and was a low cost product. Earlier, Sorizome was very costly, so it was another reason to not to pick up this market. But this product got very low cost, and it has been very, very effective. However, the company's market muscles could not match the expectations. So we went on to doing many, many work. Down the lane, I got around 30 PhDs, and through them we have generated many research work, novelty. This is one which is rated very high in collaboration with the microbiology department. We could entrap viruses, the bacteriophage, in fact, into the liposomes for the this biofilm for the biofilm, anti-biofilm activity in the wound healing and all. So we got I think 2 patents on it, and many publication, and Professor Chibber, the leading microbiologist who actually got us to do that. So we had the other molecules like methotrexate. तो बहुत सारी कहानी है, but all these stories which I see is not much. We have yet to fight a bigger battle. अभी युद्ध बड़ा है, अभी बहुत सारा अभी करना है. There is not much done. One story, two story, three story will not suffice in making our dreams, country's dreams, Make in India, Startup India, Mahan India. We are number 3 in generics, but we are 14th in values. Yet we need to go a long way in converting generics into the this innovative, the innovations. The further we have to evolve, how long we can keep our patients treating on simply on the generics. And so we have to move to the next level of efficacy and safety and compliance. There is always a scope. So we have to move forward and we have to work hard. So what did I do? The ecosystem, industry, institute, network. This is very important when you have to convert the basic research into, you have to translate the research into practical reality. So I collaborated with the multiple institutions, the government, the supported, and many colleagues, they supported wherever they it required. So it's a multi-pronged the model, not only the industry institute alone. It needs a total ecosystem. So we need to understand for this research and innovation and alluring this industry, what is with us and what actually industry need. Industry का mindset और हमारा mindset क्या है? So some of the points before you. We have in academia knowledge centric goals. हम सिर्फ यही समझते हैं कि हमें knowledge gain करनी है और वही एक गोल रह जाता है, जबकि इंडस्ट्री इज प्रॉफिट ड्रिवन. उनको सिर्फ प्रॉफिट से मतलब है. दे हैव टू सस्टेन, दे हैव टू अर्न, सो दे वांट मनी. So we need to understand and how to bridge the gap, how to convince them that there are patients, don't look to the money only. We need to treat patients. It is very difficult to convince and communicate that it is not only the profit, it is the care for the patient also that is very important. We have resource constraints. हमारे पास resources बहुत कम है. मैं देखता हूँ कि जो जितना फंड्स हमको मिलना चाहिए वो मिलता नहीं है. इंफ्रास्ट्रक्चर रिसोर्सेज जो कि फॉरेन यूनिवर्सिटीज में होता है, इसको कैसे किया जाएगा, ये दैट्स ए बिग क्वेश्चन बिफोर अस. We have to have that thinking leadership. We have long-term focus and we believe in the publications and that will in future generate some applications. So we are not looking actually for the conversion. We actually lack the industry vision कि इंडस्ट्री में प्रोडक्ट चाहिए, जबकि हमको ऐसा मतलब हमारा एटीट्यूड या हमारा जो एक माहौल है वो ऐसा नहीं है, जबकि ऐसा होना चाहिए. और we lack in effective communication, how to articulate our research findings. हमें बताना भी आना चाहिए कि हमारे उसका जो भी हमने किया है कर रहे हैं वो how it is useful, how will it be useful for the medical fraternity, medical this and industry. We have this leadership gap, mentorship, and we need who the mentors which have the industry experience. They can guide us with respect to their regulatory affairs, market-driven R&D, and incentives are also low. Why our students and PhD scholars, they are not coming to us, the talent is not coming to us because sufficient incentives are not there. We need to come to the par, at par with the world, the international standards. This is the extremely important requirement that government and the industry should understand. This is how we are losing our talent, which could be more useful. They are serving the data analytics, pharmacogenomics, and all those. They are, of course, they also need, but in research, we are lacking the real talent. And marketing and the management, that is important. But the more important is the cutting edge, mind application, your intellect. Top intellect should be attracted. This is the problem. So industry also do not like to take risk कि बहुत दिन लग जाएंगे, उनको टाइम बहुत इंपॉर्टेंट है, ट्रस्ट भी नहीं होता, डाटा इंटीग्रिटी, डाटा रिलायबिलिटी, डाटा रिप्रोड्यूसिबिलिटी इज वेरी वेरी इंपॉर्टेंट. जो हमारा एटीट्यूड है एकेडमिया में वो ऐसा ही है, चलता है, एटीट्यूड है. हमको तो जो हम एक डिग्री के लिए काम कर रहे हैं, लेकिन वो डिग्री के लिए काम नहीं कर रहे हैं, उनको तो प्रोडक्ट चाहिए, और प्रोडक्ट तब तक नहीं होगा जबकि वो प्रॉपरली उसका validation nahi hoga, until unless it passes through all the tests, the hurdles. And time is very important, and the regulatory pressures hain, competitive pressures hain. So academia should understand the need of the industry, then only we can think of, So I also collaborated in collaboration currently. We are working for multiple problems with PGI and so we need academia industry this 360 degree. So there are many things which we cannot actually talk about. But 360 degree is enough to convey the spirit कि हमारे पास में सब कुछ होना चाहिए जो होता है. और बहुत सी बातें तो ऐसी होती हैं जिनको हम एक्स फैक्टर बोल देते हैं, वो हमें पता ही नहीं होता कि क्या होता है. So what is that X factor? What are those X factors? So after narrating all this, my work experience and the stories, I'm coming to the now a very, very important dimension, which is worth talking in the light of the national education policy in the Indian knowledge system, how to create the mahal, the harmony, how to find that sanjeevani, how to create that environment, very positive energy filled environment. वो जब तक नहीं होगा जब तक कि हम अपनी जड़ों में नहीं जाएंगे. सो जड़ों में जाने के लिए हमें कैसे इकोसिस्टम क्या होता है, फॉर दैट आई ब्रॉट ए पिक्चर बिफोर यू, दैट ए रिसर्चर, ए खोजी ऑन कैंपस, वी शुड थिंक ऑफ अवर एंशिएंट इंडियन एजुकेशनल प्रैक्टिसेस, गुरुकुल परंपरा. एक्चुअली जैसे हम देखते हैं कि आर्यभट्ट, धन्वंतरी, चरक, ये सब हम इनको वैद्य के रूप में, डॉक्टर के रूप में, फिजिशियन के रूप में या एक फिजिसिस्ट के रूप में देखते हैं. बट एक्चुअली what was the system of education? In our parampara, Bharatiya parampara, the children were sent to the Gurukul, the school, to learn what? To learn what? They had only one idea in mind, ki the life ki shiksha. Even Ayurveda, they said Ayurveda is not a science to treat, but it's a science of life. जीवन का ही विज्ञान है. So it was the life, school of life. जीवन का स्कूल. ये नहीं था कि ये मेडिकल स्कूल है या इसका स्कूल है. पूरे जीवन को ही उसमें देखा जाता कि आफ्टर ऑल पूरा खोजी का एटीट्यूड यही था कि हमको ये जीवन इटसेल्फ वाज ए लाइफ इटसेल्फ वाज ए प्रोजेक्ट, रिसर्च प्रोजेक्ट. और उसको जो एक एटीट्यूड था हमारा कि हम खोज करने आए हैं कि व्हाई दिस लाइफ इज देयर, व्हाट इज द basic intent for which we are born here, why we are come here in the life. आज ये क्वेश्चन मिसिंग है. ये क्वेश्चन आज के हमें अपने एजुकेशन सिस्टम में हम बच्चे को कभी बता ही नहीं पाते. हमें खुद भी नहीं पता होता कि एक्चुअली लाइफ है क्या, व्हाट इज द पर्पज ऑफ द लाइफ. दिस इज वेरी वेरी इंपॉर्टेंट. और हम सब चीज़ों की खोज करने की कोशिश करते हैं, ये कैसा है वो कैसा है, लेकिन कभी ये नहीं सोचते कि यार हम क्या है. We are given a limited over to play in our life. Say 60, 80, ज़्यादा से ज़्यादा बंदा कितना जिएगा. 80 के बाद जी भी लेगा तो करेगा क्या. But we never had this question, who am I? Swami Vivekanand roaring with the statement कि भाई उद्देश्यहीन purposeless life. ये क्वेश्चन हमें कभी, सारे क्वेश्चन हमने सिखाए, ये क्वेश्चन नहीं सिखाया कि हमारी जिंदगी का ध्येय क्या होना चाहिए. आफ्टर ऑल हम आए कहाँ से हैं, चलो वो ना पूछो तो कम से कम ये तो पूछना चाहिए कि हमें जाना कहाँ है. जाने की तैयारी हो गई और हमें पता ही नहीं है कि हम कहाँ जा रहे हैं. जाएंगे तो हाँ भाई वो गीता में लिखा है, और लिखने से तो क्या होता है, लिखने से कुछ नहीं होता है. इससे बेवकूफ बनने वाली बात है. हमें पता तो है नहीं कि हम कहाँ जा रहे हैं, लेकिन लिखा है तो हमने मान लिया. Be fooling yourself. So this realization, this actualization, the efforts, the children were sent to Gurukul and the gurus to gain this answer. वही इसी के लिए जाते थे. Out of that, they had their own interest and hobbies. They become warrior, Arjun. They become sports person. They become Dhanvantri. They become Charak. This is very important. So they had an evolved mind, evolved consciousness, evolved their ability to think. So this sloka in the picture, what you see is very giving a very clear statement. What we understand today in drug delivery, nanotechnology, is even short of it. Because this sloka, couplet in Charak Sanhita, अल्पस्यापि महत्त्वाम प्रभुत शाल्पक मर्ताम कुर्योत संयोग विशेष काल है संस्कार है युक्त भी. मीन्स हम ये आप प्रेशर मोटर देख रहे हैं कि अगर एक छोटी क्वांटिटी को हम माइक्रोनाइज करेंगे, नैनोनाइज करेंगे, along with some excipients, meaningful excipients, and they are processed rightly, appropriately, and given to the patient with the right kind of attitude, the manobhav, and taken in the that manobhav, the shraddha, then the same chemistry, the molecule which acquired the different physics, the energy status, will actually interface with the biology and do the wonders to transform. And so this has to be understood, that how this nanomedicine and who is the father of nanomedicine. It is a clear-cut statement of Charak. He should be declared father of medicine, nanomedicine. And you see the wonder. The philosophy go hand in hand, science and spirituality. What you see is the science in this sloka. The same times it comes to the spirituality. Mahatma Gandhi said, saluting again for Mahatma Gandhi, for this time for his positive remarks, that potency of the thought lie in its subtleness. महात्मा गांधी कहते हैं कि हमारा विचार जितना सूक्ष्म होगा उतना ही हम उतना ही हम पावरफुल होंगे। जितनी हमारी ईगो डाउन होगी, कम होगी, उतना ही हम उतना ही पावरफुल होंगे। सो द पावर लाई इन ह्यूमिलिटी। ये कौन समझाएगा? आज हमारे एजुकेशन सिस्टम में तो पता ही नहीं है। ऑल योर इको सिस्टम इज गड़बड़, डिस्ट्रॉयड बिकॉज ऑफ द ईगो प्रॉब्लम। How to tame down this ego? Until unless we work on ourselves, the ecosystem, the ईगो के ऊपर काम करना बहुत जरूरी है। ये बात समझना इको सिस्टम, हमें ये नहीं कि हमें दूसरे की ईगो पे काम करना है, हमें अपनी ईगो पे काम करना है। सो दिस इन यू सी द नैनो व्हेन हमने एक पोएम लिखी है डॉक्टर गौरव, कैन आई गेट इट फॉर यू?
यस सर, यस सर।
एक नैनो के ऊपर पोयम है। वो रिवील करती है साइंस एंड स्पिरिचुअलिटी को कि नैनो का मतलब क्या है। और नैनो नैनो मेडिसिन, नैनो टेक्नोलॉजी गो विद द नैनो ईगोलॉजी, नैनो ईगोलॉजी।
मेरा लाइपोसोमल
नो नो सॉरी,
रूपांतरण।
नो नो।
एक दिन एक बेचारे नैनो नन्ही जान है पर बड़े गुणों की खान है। छोटा छोटा रूप धरती देती नई पहचान है। खोज
सर शायद आपका कनेक्शन चला गया है। डॉक्टर गीता एक बार फोन करो सर को।
Yes, sir.
डॉक्टर विकास, आपका बहुत बहुत स्वागत है। धन्यवाद समय निकालने के लिए आपका।
धन्यवाद सर। आवाज आ रही है सर?
हाँ जी, हाँ जी, आ रही है सर।
गुड आफ्टरनून सर।
गुड आफ्टरनून जी, गुड आफ्टरनून सर।
सर अभी कनेक्ट कर रहे हैं सर।
So this is very important dimension to build ecosystem. So how ये हमें Dr. Gaurav, we really need to work very hard on campus, especially leadership के लिए बहुत important है कि हम देखते हैं कि एक बार जो बॉस बन गया है ना, फिर वो बस वो बन गया। तो वो जब तक हमारा ऊपर से ही सारा गंगा comes down from top. So everywhere we see in institutions, in universities, our vice chancellors and chancellors, and there's a problem of ego, है ना। और अभी प्रॉब्लम और भी ज्यादा ये आ रही है कि जो नॉन परफॉर्मर्स होते हैं बड़े लोग और परफॉर्मर्स उनके नीचे काम करते हैं। So this how to change this the attitude of our decision makers is very difficult, है ना। हम जब वाइस चांसलर के लिए अप्लाई करते हैं तो हमारी एप्लीकेशन ना वो डस्टबिन में फेंक दी जाती है और वो पता चलता है कि कोई जो दूसरा बंदा है जिसका हम बायोडाटा मैच करते हैं तो कहीं देखता ही नहीं है। ये सच्चाई मेरी आप बीती बता रहा हूँ। और हमें वो सब आता नहीं है कि हम किसी पॉलिटिकल लीडर के पास जाए। हमारी तो जिंदगी निकल गई, लेकिन अब बच्चों के लिए क्या होगा ये हमें करना है, आगे आने वाली जनरेशन के लिए। तो इकोसिस्टम का मतलब ये नहीं है कि हमें इंफ्रास्ट्रक्चर होना चाहिए, फैसिलिटी होना चाहिए। हम एक ह्यूमन एक्सीलेंस कैसे लेके आए, और टेक्निकल एक्सीलेंस, हाउ सोवर गुड यू आर। फिर तो उसमें क्या होगा, जब तक हमारा कोहेजन नहीं होगा, ह्यूमन ईगो डाउन नहीं होगी, एक दूसरे के साथ तालमेल नहीं होगा, हम एक दूसरे को एक्सेप्ट नहीं करेंगे, टॉलरेंस नहीं लाएंगे। वेरी इंपॉर्टेंट। पहले ऐसा नहीं था। हम भारत रियली हम भारत महान थे। यू सी द राजा जनक, है ना, उनको पता भी नहीं था कि हम राजा हैं, है ना। सारी चकाचौंध से दूर थे वो। राजा होते हुए भी उन्हें पता नहीं था कि हम राजा हैं। वो सिस्टम आप कैसे लाएंगे, उसका एक ही तरीका है, बुद्धम शरणम गच्छामि। मेडिटेशन इज द मंत्र। इट इज ओनली इन मेडिटेशन वी वर्क ऑन आवर ईगो। हाउ? जब आप मेडिटेट करते हैं तो बुद्धम शरणम गच्छामि, माने आप अपने को सरेंडर करते हैं। और वो सरेंडर करने का मतलब ह्यूमिलिटी क्रिएट करते हैं कि हे ईश्वर, जो भी है आप हो। आई एम दी ट्रांसफॉर्म ही इनटू पॉजिटिव पर्सन। So this meditation is very important, where one is allowed to learn surrender, processing. अदरवाइज तो हम हमेशा यही समझते हैं कि यार दूसरा नेगेटिव है, मैं ही पॉजिटिव हूँ। सब हमेशा when you think of introspection, people always think introspection for others. So meditation is the only way where you introspect and correct. There are two things. When you close your eyes, just make an experiment and you close your eyes, you will find lots of things which are not you are not happy with about yourself. So you will find lot many things which you need to work on, to correct, to improve. तो आपका जो सेल्फ डेवलपमेंट है, पर्सनालिटी डेवलपमेंट है, वो कैसे होगा जब तक कि आपका कैरेक्टर फॉर्मेशन के जो फैक्टर्स हैं वो आइडेंटिफाई नहीं होंगे। So Buddham Sharanam Gacchami. Now what I see as another interesting picture, the current state of mind, because मैं चाहता तो हूं लेकिन हो नहीं पाता है, ये स्टेट है डिसऑर्डर्ड स्टेट, फ्रेगमेंटेड माइंड। एक गीता में बहुत अच्छा जो है, आपने भी पढ़ा सुना होगा, जानामि धर्मम न चा मैं प्रवृत्ति। मैं दुर्योधन कहता है मैं जानता हूं धर्म को, लेकिन मेरी प्रवृत्ति नहीं होती। आई हैव नो प्रोपेंसिटी टुवर्ड्स द राइट थिंग्स। जानामि धर्मम, मैं धर्म को जानता हूं, लेकिन मैं क्या करूं, मैं खुद ही के बस में नहीं हूं। जानामि अधर्मम न चा मैं निवृत्ति, मैं जानता हूं अधर्म क्या है, मैं करता हूं अधर्म, लेकिन मेरी उससे छुटकारा ही नहीं मिलता मेरे को। वाई? बिकॉज दिस माइंड इज एक्चुअली द ग्रेट थिंग। इट इज सो in the state that it creates a lot of problems. So you need for your leadership excellence, research, innovation, tech transfer, सबके लिए माइंड ही तो डायनामो है। तो mental efficiency index is down. Only through meditation, which I practice, the heart-based meditation, where you meditate on heart, because heart has a bigger role as a actual very important thing which remain unexplored hitherto. So we create that ordered state uh, so that your efficiency, your mental sharpening takes place. So what difference between the non-meditative mind and mind is you एक ही लाइन में but it thinks at 360 degree. मतलब ये स्टूडेंट्स वाले को केमिस्ट्री देखेगी, केमिस्ट्री वाले लिवर को देखेगा। तो ये जो थ्री जो आपका माइंड योगा की भाषा में बोलते हैं कॉन्शियसनेस, चेतना, अवेयरनेस, that is a very very important not only for spirituality, it is important for science also, because तब तक आप नहीं कर पाएंगे, एक ही डेटा कर रहे हो, डेटा नजर में ही नहीं है क्योंकि आपका उस तरफ ध्यान में ही भगवान ने इस probably I am हाँ प्रेजेंटेशन में आ रहा है, कोई बात नहीं, छोड़ दो उसको। So the meditative mind and non-meditative mind, there is a lot of turbulence there. You can understand the rheology, the Reynolds number, the unit operations that we learned. So जब mind अगर still है, एक current उसकी turbulence नहीं है, non-turbulent है, then you have the better ability to create innovation and to create the atmosphere, to lead, to join hands, so much so. So the science and spirituality go hand in hand. Material progress and spiritual progress, they are the two wings. Both are necessary. पहले बहुत सब कुछ स्पिरिचुअल करते थे, हमारे संत महात्मा जंगल में चले जाया करते थे स्पिरिचुअलिटी के लिए। लेकिन ये राजा जनक मॉडल में आपको दिखा रहा हूँ कि राजा जनक हमारे इतिहास में एक ऐसे ऋषि हुए हैं जो राजा भी थे, वो ऋषि भी थे। तो हमारा मॉडल जो है ना, हमारा इकोसिस्टम भी इसी फिलॉसफी के साथ ड्रिवन होना चाहिए। हमें सिर्फ सन्यासी नहीं बनना है, ना ही हमें हमें घर गृहस्थी में रह के, फैमिली लाइफ में रह के पूरा युद्ध पराक्रम करते हुए सब रिसर्च, इनोवेशन, टेक्नोलॉजी ट्रांसफर और सब कुछ करना है। So this is a Raja Janak model. So we provide 4 practices: heartfulness way, and very simple techniques. I think your campus is Heartful campus, and they are conducting all these sessions, Heartful campuses. Very simple practices, but very effective. And कोई भी इसमें वो नहीं है, ritual नहीं है, mechanics नहीं है. It is all organic. So this is all cost free. You can download the app and get to the online trainers, and physically also people like me, 16,000 trainers all over the globe are available to take care for the one-on-one meditation free of cost. So this is one thing I want to highlight. I had many recognitions. I served almost all positions on the university. One is a chairman, dean, and director research. It is all because of the research and innovation, the actual practical reality that it got through to the practical these patients and the market, and we earned revenue back for the university. That's also a rare in the form of the royalty. So many recognitions. The president award, कलाम साहब ने बुलाया, हाथ मिलाया और 1 लाख रुपए भी दिया था 2007 में. और उसके बाद में इंटरनेशनल रिकॉग्निशंस, आई वाज इनवाइटेड मल्टीपल टाइम्स टू द फास्टफूड लिपिड रिसर्च सेंटर जर्मनी. आई एम द फाउंडर मेंबर देयर एंड आई हैड ए मेनी अवार्ड्स. इसमें भी 1 लाख रुपए मिला था. और story goes on. Our students also earn a lot of this now. Dr. Kesar Raza and many students, now 30 scholars, PhD are almost, they are serving top positions in the industry. We have a recent launch by IPCA Labs doing good in the market. We have another of the methotrexate gel by IPCA that is also doing good. So हमने हार नहीं मानी, हम आगे बढ़ते चले गए. अभी जल्दी ही आप देखेंगे 3-4 प्रोडक्ट और हमारे पाइपलाइन में जल्दी आ जाएंगे बाहर. So we got this award more than my science and technology award. I rate it very high, which I got for my self-development work for the youth empowerment at Center for Swami Vivekananda Studies. This heartfulness is all about the for Swami Vivekananda philosophy. So it is closely aligned, or rather it emanates from that philosophy of Swami Vivekananda. The self-development means you have to develop from within. So we had this, the success recipe, formula for success, that we have to have the Arjun जैसा लक्ष्य, ध्रुव जैसी प्रतियोगिता. मैंने हमें खुद को जानने के लिए, हु एम आई के लिए बुरा होने की जरूरत नहीं है. जैसे ध्रुव ने पूछा था क्वेश्चन, वैसे हमें बचपन से ही क्वेश्चन हमारे बच्चों के दिमाग में आना चाहिए कि आई हैव टू who is God actually. I'm not happy with the simple answer, God is there, but I need to know who actually, where he is. And a realization, the self actualization is very important. Otherwise जो नास्तिक हैं वो बेहतर हैं आस्तिक से, क्योंकि they are honest actually. They say God is not there, actually honesty is there in that. But we are fooling ourselves, God is there, but where is the God? All these kachra, all these jealousy factors, we all keep it, and where is the God? So until unless that happens, so भागीरथ प्रयत्न, गंगा को नीचे लाना था तो भागीरथ आए. हम भी एक ड्रग नीचे ला सकते हैं और अपने इकोसिस्टम बना सकते हैं. Devotion is very important. It's all about humility, because the moment you do something good, you host ego. So this ego, sometimes मैं कहता हूँ कि जो चोर है वो ईमानदार से ज्यादा अच्छा है. कि चोर वाले को पता रहता है कि यार मैंने चोरी की है और वो हम्बल रहता है, लेकिन जो ईमानदार है वो अपने घमंड में घूमता है कि मैं ईमानदार हूँ. तो उसके ईगो को अपन नहीं टैकल कर पाते हैं. एक जो बेईमान होता है जो, तो वो उसको कम से कम ये रहता है कि यार ये है और मुझे ट्रांसफॉर्म होना है. वेरी इंपॉर्टेंट दिस aspect. So devotion is very important. So this is a simple formula for all our youngsters to look at. This is the end of the session. Thank you so much.
Thank you very much, sir. When I heard about that there is a lecture of आपका lecture है मतलब, तो मुझे सबसे ज्यादा वेट इसी चीज की थी कि मैं जैसे वो कहते हैं ना मोटिवेशनल टॉक, तो मैं गीता मैम से यही कह रही थी, मैम सर का ना मोटिवेशन पे भी एक लेक्चर रख देंगे। बिकॉज आपका पहले भी हुआ था हमारे कॉलेज में लेक्चर, वर्चुअली था दैट टाइम ऑल्सो। तो जो रिसर्च पार्ट है that is okay, but the motivation that you give us to do something better and to think in the right direction with the right mindset, that is very important for us. मतलब कोई भी चीज है, हम यहाँ पे जैसे रिसर्च भी करते हैं, कुछ भी करते हैं, जैसे आपने कहा कि हम पब्लिकेशन तक रुक जाते हैं बिकॉज देयर आर सम कंस्ट्रेंट्स दैट academics institutions are getting in the way, and most importantly the gap between the industry and the academia, that is also maybe ये भी शायद one of the important thing है। तो थैंक यू वेरी मच सर। और हार्टफुलनेस ऑर्गेनाइजेशन सर, हम उनकी वर्कशॉप्स यहाँ पे कंडक्ट करवाते हैं सुधीर सर के साथ, तो वो बहुत ही पीस गिविंग है। मतलब हमें बहुत सूदिंग लगता है, हम ऐसे नहीं सोचते कि हाउ वी आर कनेक्टेड टू द नेचर एंड ऑल, बट आफ्टर लिसनिंग टू दैट मतलब मैं उस डायरेक्शन में शायद सोचने लग गई हूँ। तो इट्स वेरी गुड थिंग,
अगर
अदरवाइज
आप
मुझे नहीं पता था कि ये पेड़ पौधों के साथ भी हम कनेक्टेड हैं, या फिर जो आपस में ह्यूमन बींग्स आर कनेक्टेड टू द इंसेक्ट्स ऑल्सो, मतलब एवरीथिंग इट्स ऑल वन थिंग, वन इकोसिस्टम। So they are also very good lectures there that are conducted over here. So thank you very much, sir.
एक्चुअली अगर हमें एक्सीलेंस लाना है, और इट्स ए टाइम ऑफ दिस कॉम्पिटिटिव वर्ल्ड, है ना, तो आगे आने वाले टाइम में आप देखेंगे कि डेफिनेटली इट विल मेक ए डिफरेंस द मेडिटेशन। व्हाट, जो मेडिटेटिव माइंड है और जो नॉन मेडिटेटिव माइंड है, अगर आपके कैंपस में 10 मेडिटेटिव माइंड इकट्ठे हो जाते हैं तो यू कैन डिफीट पंजाब यूनिवर्सिटी। आई मीन इट। यू कैन एक्चुअली, जो भी मैं बात कर रहा हूँ दिल से कह रहा हूँ। अगर 10 मेडिटेटिव माइंड भी इकट्ठे हो जाते हैं, 10 से 100, 100 से 1000, तो वो क्या होता है, एग्रीगोर बनता है वहां पर, एक माहौल ही अलग बन जाता है, आप कुछ भी कर सकते हैं। तो मेडिटेशन इज वेरी वेरी इंपॉर्टेंट। वी शुड नॉट साय, वी शुड नॉट हेजिटेट टू टेल अवर यंगर जनरेशन। और इसके अलावा यही नहीं प्रियंका जी, अभी यंगस्टर्स को अपनी फैमिली करनी है, अभी शादी करना है, किससे करना है, वो लड़का कौन सा पसंद होगा, क्या करेगा। वी नीड क्लैरिटी ऑफ द माइंड। क्लैरिटी ऑफ माइंड कैसे आएगी? क्लटर्ड माइंड कैन नॉट टेक डिसीजंस, और लेगा भी तो गलत ले लेगा। तो अभी कोई भी डिसीजन मेकिंग है, चाहे लीडरशिप हो, चाहे पेटेंट बनाना हो, चाहे शादी करनी हो, चाहे फैमिली चलानी हो, आपको मेडिटेटिव माइंड तो चाहिए ना। यू शुड हैव दिस स्पिरिचुअल कॉर्ड, देन यू डिसीजंस विल कम बेटर। आपको जो है ना, विवेक, बुद्धि से विवेक की तरफ जाना है। बुद्धि तो भगवान ने हम सबको दी है, पर विवेक क्या होता है? विवेक ऐसे नहीं मिलेगा, विवेक अपने ऊपर काम करके ही मिलेगा, फ्री में नहीं मिलेगा। तो बुद्धि से विवेक की तरफ जाना है, अहम को छोटा करना है और चेतना को बढ़ाना है। एक्सपेंशन ऑफ द कॉन्शियसनेस। मन, बुद्धि, चित्त और अहंकार, ये हैं व्यक्तित्व के 4 तार, है ना। दीज आर द 4 wires, rings of the personality. So it's a grace that you have the heartfulness on your campus. I wish that you make your campus the heartful torch bearer in the region. And अगली बार आप हमें फिजिकली बुलाइएगा तो हम आएंगे।
Sure, sir.
Definitely, sir.
वर्चुअल में।
Thank you very much, sir. I
Thank
think
you.
your legacy is very clear for everything. That is the hope, progress and cheer. जब आपका सेशन हम सुनते हैं तो उसमें हमें होप भी दिखती है, प्रोग्रेस भी दिखता है और चेयर भी दिखता है कि हाँ कुछ हुआ है, शायद तो हम भी कुछ ऐसा कर सकते हैं। हम पंजाब यूनिवर्सिटी को बीट तो ना कर पाए, लेकिन जहां से पंजाब यूनिवर्सिटी ने अपनी जर्नी शुरू करी है, अगर हम वहां भी पहुंच पाए ये सब करके, तो हम अपने आप को भाग्यशाली समझेंगे सर। थैंक यू वेरी मच फॉर वंस अगेन गिव योर टाइम टू अस सर। थैंक यू वेरी मच सर।
Thank you.
थैंक यू सर, वेरी मच सर।
My gratitude and the pleasure to the institute, to all of you for giving me the opportunity to share my heart. Mind तो हम शेयर करते ही रहते हैं, है ना? थैंक यू।
Thank you so much, sir.
थैंक यू सर।
Dr. Priyanka, please go ahead.
Now, I would like to introduce our next speaker of the day, that is Dr. Sudhir Bhardwaj, Director, Divine International College of Pharmacy, Gwalior, Madhya Pradesh. He is Vice Chairman of Association of Registered Pharmacists on national level and also having responsibility of Vice Chairman Indian Pharmaceutical Graduate Association, IPGA of MP state. And he is pharmaceutical teacher and researcher having more than 20 years of PG and UG teaching experience. His work experience is in the development and evaluation of conventional dosage forms, novel drug delivery system, and microwave synthesis techniques. He has received Young Scientist Award twice at international and national conferences and has published more than 50 research and review articles in different international and national journals. He has presented more than 25 research articles in oral and poster presentation and published 4 books in the field of pharmaceutical sciences. So I welcome you, sir.
Thank you, ma'am. I'm audible for everyone?
Yes, sir. Yes, sir.
Okay. So first of all, very good afternoon, everyone. And I'll just congratulate everyone and especially entire team of GGS COP. They have just organized this webinar or make available a platform for deliver some views. Actually, now I don't have anything to share because you have already invited people from the industry and the different institute. So the exact knowledge training has been completed. They have just delivered all the aspects of research and the things that is exactly required for the academics. So, but I'll just complete my responsibility as you invited me. So it's really an immense pleasure for me. I also, thanks to all the student, young leaders, and all the participants, they are here. And I'll not take a more time. I'll just make a very comfortable presentation, or even I'll say I'll just share my views, not coming with a presentation, just sharing my view. One thing that I would also like to say that you really selected a very good speakers because if you are inviting a people from the higher or the institute where already research has been completed by the senior professors, so they would have a capacity to deliver the thing. Even they can give a learning to us that how to work with a minimum resources and how we can get a maximum output on that. क्योंकि किसी भी संस्था में यदि if senior teachers they have done some work that was done by the very minimum resources, आज हमारे पास जो संभव संसाधन है वो तब नहीं थे. तो ये आपका बहुत अच्छा फैसला था. आई एम रियली अप्रिशिएट दिस इज रियली वेरी गुड एंड द वे दे हैव गिवन द all the important part that's related to the fostering of research and innovation. देखिए जब हम फॉस्टरिंग की बात करते हैं तो वो फॉस्टरिंग तभी हो पाती है जब हम उन लोगों के बीच अपनी बात रखें जिनको वाकई उस चीज का मतलब पता है. हम प्रोत्साहित किसको करेंगे? हमारा प्रोत्साहित करना तभी पॉसिबल हो पाएगा जब वो लोगों के साथ हम प्रोत्साहित करें जिनको वाकई ही जरूरत है उस चीज की. एंड दैट्स आई थिंक कम विद अ बैचलर, इवन 11th 12th student, intermediate student, they are basically our object for us if we really want to make the things comfortable for us. क्योंकि बिगनिंग से जब तक बच्चे का माइंडसेट नहीं बनेगा रिसर्च का, इनोवेशन का, उसको पता ही नहीं होता कि हमें जाना कहाँ है. और जब वो वहां आ जाता है तो हमारे पास बैचलर एक ऐसा प्रोग्राम है जहां की हम चीजों को बता पाते हैं. So, Dr. Gaurav, the topic of this whole theme is very good. एंड जो मुझे लगता है जिन चीजों के लिए मैं अभी जो आपसे शेयर करना चाहता हूं या जितने भी पार्टिसिपेंट हैं उनके साथ में शेयर करना चाहता हूं, तो मैंने कुछ पॉइंट्स बनाए हैं जिसमें कुछ पॉइंट्स ऐसे हैं जो हमारी रोजमर्रा की जिंदगी के लिए, जो डे टू डे लाइफ में हम देखते हैं कि इफ वी आर अ टीचर, सो वी वुड हैव सम डिफरेंट काइंड ऑफ challenges. If the people they are listening me, they are a student, they are having a different kind of problem or obstacles they are facing every day. So student और टीचर, रिसर्चर की और इनोवेशन, जो रिसर्चर्स जो इनोवेट कर रहे हैं कुछ, उनकी हम बात ही नहीं कर रहे. वी आर जस्ट वेरी मच कनेक्टेड टू द पीपल, दे आर वर्किंग फॉर द टीचिंग एंड दे आर टेकिंग अ स्टडी फ्रॉम द इंस्टीट्यूट. अब बात आती है इंस्टीट्यूट की. इंस्टीट्यूट फॉर व्हाट? As you know that GGS COP is doing a very well work because they are delivering a basic information, basic curriculum with a B.Pharm course, but simultaneously they are working for the research. अब जब रिसर्च को आप डिलीवर कर रहे हैं तो आपकी एक टीम बनी हुई है. उस टीम में जो अभी सर बार बार बोल रहे थे कि एक इकोसिस्टम डेवलप्ड है. वो इकोसिस्टम इसलिए बना हुआ है क्योंकि जब पॉजिटिव माइंडसेट के साथ हम कोई चीज करते हैं तो नॉलेज भी डिलीवर हो पाती है, और जो लोग हमने वहां पर रिसर्च को नर्चर करने के लिए रखा है या रिसर्च को बताने के लिए रखा है, वो भी अपनी बात को सही ढंग से रख पाते हैं. तो इंस्टीट्यूट इज बेसिकली होता क्यों है? इंस्टीट्यूट इज जस्ट फॉर एक दो, मुझे लगता है कि दो मेन कारण है कि जब किसी इंस्टीट्यूट में, कॉलेज में, स्कूल में जब कोई बच्चा जाता है, स्टूडेंट जाता है, तो उसके लिए सबसे पहली चीज होती है डिसिप्लिन सीखना, और दूसरी चीज होती है बेसिक नॉलेज को कलेक्ट करना. यहाँ पर रिसर्च की बात करें या हम इनोवेशन की बात करें, ये सारी चीजें हमारी बाद की बात है. बट दो चीजें जो मुझे लगता है वो सबसे पहली है, एक डिसिप्लिन और दूसरा है उनकी बेसिक नॉलेज जो इनहांस होती है उस जगह पर. अब हम बात करते हैं एकेडमिक्स की. आप देखिए, if any industry is working for the society, they should require a skilled people. They require a people they are working for exact that particular work. बट वो इंस्टीट्यूट के साथ कोलैबोरेट नहीं कर पाते हैं. जैसे सर ने भी अभी बोला कि दे आर जस्ट वरीड फॉर देयर प्रोडक्ट एंड दे जस्ट वांट टू कम विद अ सेल. लेकिन जो स्टूडेंट है वो आता है हमारी इंडस्ट्री के लिए, और इंडस्ट्री सर्व कर पाती है सोसाइटी के लिए. अगर इंडस्ट्री और स्टूडेंट और एकेडमिया के बीच में कोरिलेशन डेवलप करना है तो उसके लिए सबसे इंपॉर्टेंट चीज है माइंडसेट. वो माइंडसेट तभी बन पाएगा तब हम बच्चे को वो उस इन्वायरमेंट में भेज पाएंगे. देखो, अब इंडस्ट्री हमारे पास चल के नहीं आएगी. ये आप ऐसा मान लीजिए कि उनको हम उतना शायद नहीं दे पा रहे हैं, ऐसा मान लो, क्योंकि जो हमारा सिलेबस है, जो इंडस्ट्री की रिक्वायरमेंट है, वो डे टू डे ऐसा कह सकते हैं कि हर दिन बदल रही है. इवन विद एवरी सेकेंड बदल रही है. और जब उनकी रिक्वायरमेंट हर सेकेंड बदलेगी या उनका अपग्रेडेशन हर पल बदलेगा, तो इंस्टीट्यूट कैन नेवर फुलफिल दीज काइंड ऑफ थिंग्स. तो हमें सबसे पहले जो ध्यान रखना है वो ये रखना है कि बच्चा कैसे भी करके इंडस्ट्री के साथ इन्वॉल्व हो, चाहे वो ट्रेनिंग वाले पार्ट पर हो, चाहे वो किसी उनको हम अपने यहाँ इनवाइट करते हैं, जैसे आज आपने जो मैडम को इनवाइट किया था, उनके जो प्रेजेंटेशन में चीजें थी. बट वो चीज अगर डेवलप हो पाती है तो डेफिनेटली चीजों को हम उस तरह से प्लान कर पाएंगे या हम डिलीवर कर पाएंगे. तो अगर सर्विंग के लिए स्टूडेंट तैयार करना है तो इंडस्ट्री को भी हमारे साथ अलाइन करना पड़ेगा. चीजों को उसी तरह से हमें प्रेजेंट करना पड़ेगा जिस तरह से बच्चा आज हमारे लिए बन के तैयार होगा. देखिए, आज हम जिन जॉब्स की बात कर रहे हैं, वी आर इन द 2025, but in 2030 or in 2035, what do you think, which kind of a job would be available for the student? Because the person, the student, they are coming to take an admission in 2025, they'll going to take they'll come for the job in 2029. But every day, especially that era, that I would say this the corporate era, they are totally going for the AI. They are totally taking they are taking a support from the AI. And even you'll be surprised to know, even I think कि we are not with the technology, we are comparing with the technology. These are the instruments to enrich ourselves. These are the instruments, they'll make the process easier for me. और हमने क्या किया है, हम डिपेंडेंट हो गए हैं उनके. So we have to change ourselves. We have to redefine the exact purpose of the education. और एजुकेशन का पर्पस तभी रिडिफाइन हो पाएगा जब हमें ये पता रहे कि हम किस चीज के लिए काम कर रहे हैं। तो चाहे हम रिसर्च को फॉस्टरिंग करें या हम इनोवेशन की बात करें, एकेडमी या इंडस्ट्री की बात करें, अल्टीमेटली कॉलेज इंस्टीट्यूट इज द ओनली वे जो आने वाले समय के लिए लोगों को तैयार कर पाएगा। क्योंकि एज ए टीचर दैट्स आवर रिस्पॉन्सिबिलिटी कि हम उनको, अपने बच्चों को, अपने रिसर्चर्स को, अपने कलीग्स को या हमारे साथ जो यंग लीडर्स डेवलप हो रहे हैं उनको हम कैसे अच्छा कर सकते हैं। तो वो तभी पॉसिबल है जब हम आज डे टू डे लाइफ में हम चीजों के साथ कनेक्ट करेंगे। रिसर्च के दो पार्ट बहुत इंपॉर्टेंट पार्ट होते हैं, एक माइंडसेट होना और दूसरा होता है कि आप सोशल इंटरेक्शन के साथ कितनी चीजें निकाल के लाते हैं। देखिए अभी भी हम जो रिसर्च कर रहे हैं, अभी भी हम देखते हैं कि हम जनरली लैब की रिसर्च को आगे प्रोसीड करना चाहते हैं, हम लैब की रिसर्च को बात करना चाहते हैं, बट हम सोसाइटी के साथ कनेक्ट होकर रिसर्च को नहीं करना चाहते। सोशियो इकोनॉमिक इम्पैक्ट के बारे में हम बात नहीं करना चाहते, या करते भी हैं तो वो बात इतनी होती है कि उस बात को हम कितने सही ढंग से ले पा रहे हैं वो हमें नहीं पता होता। तो यदि हमें वो चीज करनी है तो हमें ये ध्यान रखना पड़ेगा कि सोसाइटी के अंदर जो प्रॉब्लम है, क्योंकि रिसर्च का केवल एक ही पार्ट नहीं होता कि हमने प्रोडक्ट डेवलपमेंट में चले गए। एज ए फार्मासिस्ट हमारी रिस्पॉन्सिबिलिटी बहुत अलग अलग है। एक तरफ हमें टीचर भी रोल प्ले करना है, एक तरफ रिसर्चर भी रोल प्ले करना है। यदि जो लोग मेडिकल स्टोर पे काम कर रहे हैं तो वो हॉस्पिटल फार्मेसी को भी हेल्प कर रहे हैं, पेशेंट केयर भी हमें ध्यान रखना है, पेशेंट कंप्लायंस भी हमें ध्यान रखना है। तो हमारा जो स्पेक्ट्रा है वो बड़ा है और उसके लिए हमें स्टूडेंट की नीड होगी कि हम कैसे स्टूडेंट को वहां से निकाल के लाएं जो उस पर्टिकुलर माइंड सेट के हैं, जो उस पर्टिकुलर काम के लिए बन पाए। बिकॉज दिस इज नॉट द वे कि द पीपल दे आर कमिंग, वी आर प्रोवाइडिंग अ प्रोजेक्ट, वी आर गिविंग अ टॉपिक एंड डू इट। yes, that may be possible, but the way that would be totally dependent system, or I would say the research basically work with the methodological aspects and the systematic aspects. मैथोडोलॉजिकल मतलब हमने कोई मेथड बनाया और सिस्टमिक जो जो जिस तरह से हम आगे चलेंगे, ये एक रिसर्चर का माइंडसेट होता है। और रिसर्च भी दो तरह की होती है, एक तो कोरिलेशन रिसर्च और एक अपनी जो होती है एक्सपेरिमेंटल रिसर्च। तो अगर हम कोरिलेशन की बात करेंगे तो कोरिलेशन कौन से बच्चे ऐसे हैं जो बायोस्टैटिक्स को जानते हैं, स्टैटिक्स को जानते हैं, किस ऐसे बच्चे का पार्ट है जो उस पार्ट पे मैथमेटिकली इनरिच है, तो वो वहां पर अपना रोल प्ले कर सकता है। दूसरा यदि हम बात करते हैं एक्सपेरिमेंटल पार्ट की, तो एक्सपेरिमेंटल पार्ट में जो कुछ भी है वो हाइपोथेसिस हमारी तभी कंप्लीट हो पाएगी जब उस हाइपोथेसिस में हमारे जो हमने जो मेजर्स किए हैं या जो हमने रीडिंग्स ली हैं, जो हमने उसके अंदर पॉइंट्स निकाले हैं, उनको हम कंप्लीट कर पाए। अब देखिए साइंटिस्ट जो होते हैं जब भी कोई काम करते हैं तो उनकी जो ऑब्जर्वेशन होती है वो ऑब्जर्वेशन उनके हाइपोथेसिस को कंप्लीट करता है, जो भी वो हाइपोथेसिस डेवलप करते हैं और उस हाइपोथेसिस को वो कंप्लीट करके बताते हैं ऑन बिहाफ ऑफ रिजल्ट। अब चाहे उसमें बेसिक लेवल पे उन्होंने रिसर्च किया हो, चाहे अप्लाइड लेवल पे किया हो या पेटेंटिंग पर भी किया हो, जैसे आज पेटेंटिंग की सर बात करें, बट पेटेंटिंग होगी कब? पेटेंटिंग तभी होगी जब वो बाकी सोसाइटी के लिए रिक्वायर्ड है या बाकी सोसाइटी के लिए जरूरी लगती है, अदरवाइज फिर उस पेटेंटिंग का कोई मतलब ही नहीं होता है। तो रिसर्च को यदि हम देखते हैं तो मुझे जो लगता है, अगर मेथोडोलॉजी की बात करते हैं तो रिसर्च मेथोडोलॉजी में भी सबसे इंपॉर्टेंट पार्ट आता है, वो तो है लिटरेचर रिव्यू। हम लिटरेचर को उतना नहीं कर पाते अभी, तो फिर बहुत सारे साधन आ गए हैं। आज आप ऑनलाइन इस युग में हो, वर्चुअल वर्ल्ड में आप बैठे हुए हैं, बट फिर भी हम इसलिए नहीं समझ पा रहे हैं या हम बच्चों को वो चीज नहीं दे पा रहे हैं कि किस किस एस्पेक्ट से उनको देखना है। एक मोबाइल है, अगर हम इसको देखते हैं, ऐसे भी देख सकते हैं, ऐसे भी देख सकते हैं, ऐसे भी देख सकते हैं, तो वो कब पॉसिबल हो पाएगा? वो तभी पॉसिबल हो पाएगा जब हम उनको ये सिखाएंगे कि एग्जैक्टली लिटरेचर रिव्यू का एस्पेक्ट्स क्या है। अब लोग पेपर निकालते हैं, टीचर ने प्रोजेक्ट बताया, उसने उसी के आधार पर बनाया, उस बच्चे को तभी डेवलप हो पाए। अभी हम क्या कर रहे हैं, हम प्रोजेक्ट को कंप्लीट करना सिखा रहे हैं और हमें सिखाना क्या है कि वो कैसे रिसर्च को कंप्लीट करे। तो रिसर्च सीखने के लिए उसको केवल उस मेथोडोलॉजी का पार्ट बताना है, बाकी वो अपने आप कर लेगा। तो मेरे जो व्यूज थे या जो चीजें, मैं चूंकि बहुत सारी स्लाइड्स बनाई थी, बट वो स्लाइड अब मैं इसलिए भी नहीं शेयर करना चाहता हूँ क्योंकि सारी जो सबकी लिसनिंग पावर है ना, वो सब खत्म हो चुकी है। We have already taken around the 3 hours classes because you started around 11 and that was around 1:30. So I'm just telling you as a teacher, I think most of the participants are from the teaching side, na? Or some of you will be a student. हमें उन चीजों पर तो ध्यान रखना पड़ेगा जो बेसिक्स हैं और बेसिक यही है कि स्टूडेंट के साथ उसको माइंडसेट के अकॉर्डिंगली उसको तैयार करना। जो आज एआई के ऊपर डिपेंडेंसी बढ़ती जा रही है उस एआई को भी पढ़ने का तरीका देखिए। कंप्यूटर सारे काम करता है लेकिन कंप्यूटर बनाया किसने है? वो तो ह्यूमन ने ही बनाया ना। और ह्यूमन ने कब बना पाया? जब उसने सोसाइटी की और वर्किंग की नीड को समझा। तो कंप्यूटर वुड बी योर फ्रेंड बट दैट कैन नॉट बी योर फादर। अब हो क्या गया है, हमने उसको खुद को ही इतना ज्यादा तवज्जो दे दी है कि हर चीज उसी के आधार पर चल रही है, हर काम हम उसी के अकॉर्डिंग करना चाहते हैं। तो उसकी हेल्प हो और बच्चों को भी ये समझ में आना चाहिए कि यदि हम वहां से कोई चीज ले रहे हैं तो वो सही और गलत में कैसे अंतर करें। और वो तभी हो सकती है जब आपके पास बेसिक लर्निंग होगी, बेसिक आपके पास कहना चाहिए कि सीखने की वो बात पता होगी कि हमने किस तरीके से आगे तक इस चीज को लेके जाना है। एक चीज और आती है, रिसर्च में टीम बिल्डिंग बहुत इंपॉर्टेंट पार्ट प्ले करती है। If we want to nurture a research, we just want to innovate something. So टीम बिल्डिंग से मेरा एस्पेक्ट है कि यदि आप टीम बिल्डिंग में जाना चाहते हैं तो आपको ये देखना पड़ेगा कि एक्सपेरिमेंटल पार्ट में कौन अच्छा है, स्टैटिक्स को कौन यूज अच्छे ढंग से कर रहा है, प्रैक्टिकल जो कंफर्मेशंस हैं या प्रैक्टिकली हम जो चीज कर रहे हैं उसको अगर हम रिव्यू करते हैं तो ऐसे कौन है ऐसा स्टूडेंट या टीचर या हम उनसे उस चीज के बारे में बात करें, हम उनसे उस चीज के बारे में सीखें। होता क्या है, जब हम बहुत सारी चीजें एक साथ सीखना चाहते हैं, इफ वी आर टेकिंग अ नंबर ऑफ थिंग्स इन डिफरेंट वे, तो रादर देन दे आर गोइंग टू इन योर माइंड, दे विल मेक यू सम इनोसेंस इन योर माइंड। दे आर नॉट गिविंग अ एग्जैक्ट इंफॉर्मेशन, दे आर जस्ट मेकिंग अ इनोसेंस। क्योंकि आप बहुत सारा जब ज्ञान ले लेते हैं हर चीज को आप अच्छा नहीं कर सकते। और जब हर व्यक्ति उस तरह से होगा, जैसे अभी जो सर ने भी बोला था अभी कि हम नई एजुकेशन पॉलिसी के बात कर रहे हैं, वो पॉलिसी जब आएगी और उसके क्या एस्पेक्ट्स होंगे क्या होगा पता नहीं। बट अगर 2 बच्चे ऐसे हैं, 3 लोग ऐसे हैं, वो 3 लोग मिलके भी अगर एक काम करते हैं तो वो भी एक अच्छा पार्ट हो सकता है नर्चरिंग करने के लिए, फॉस्टरिंग करने के लिए रिसर्च को और इनोवेशन। इनोवेशन होना या रिसर्च करना, रिसर्च में तो हम मानते हैं कि आप अगर चाहे बेसिक रिसर्च कर रहे हो, चाहे मॉडरेट कर रहे हो या पेटेंटिंग पे भी जा रहे हो, पेटेंट कंघे तक का हो रहा है लेकिन वो कंघा काम का है। आप देखिए रोज कितनी सारी क्रीम्स आ रही है, फार्मेसी के अलावा सारे लोग क्रीम बना रहे हैं, तेल बना रहे हैं, पता नहीं वट एवर यू विल जस्ट कम टू द दिस इंस्टाग्राम, everybody is making a drugs and the different kind of a doses form and they are doing the patent. Why they are going towards patent? Because उनका जो प्रोडक्ट है वो कहीं ना कहीं सोसाइटी को रिच कर रहा है, सोसाइटी की हेल्प कर रहा है, या सोसाइटी को हम कह सकते हैं कि चीजों को बताने के लिए तैयार है। दूसरा, इंडस्ट्री के साथ अगर हमारे कुछ कोलैबोरेशंस होते हैं या हम जा पाते हैं तो सबसे बड़ा पार्ट होता है वहां का एनवायरनमेंट, विजुअल। मुझे जहां तक लगता है और ये बाकायदा अप्रूव्ड है, ये चीज ऑथेंटिक है, यदि विजुअली आप किसी चीज को देखते हैं तो हमारी मेमोरी जो होती है वो लगभग 90% उस चीज को याद रखती है। हम जनरली मूवी देख के आते हैं तो आपने देखा मूवी हमें कई दिनों तक याद रहती है। यदि हम और भी किसी चीज के बारे में बात करते हैं, हम किसी सिटी को घूम के आए हैं तो हमें पता होता है कि हम कहाँ गए थे, कितनी देर के लिए गए थे। तो ये सारी चीजें हमें पता होती, क्यों? बिकॉज आवर मेमोरी इज ग्रास्पिंग, दे आर मेकिंग अ पिक्चर्स एंड दे आर स्टोरिंग द पिक्चर इन आवर माइंड। तो अगर बच्चा बैचलर्स में इंडस्ट्री के साथ कोलैबोरेट करता है, वहां का एनवायरनमेंट देखता है, तो हो सकता है उसके माइंडसेट में वो एनवायरनमेंट सम कुछ पॉजिटिव माइंडसेट को डेवलप कर पाए और पॉजिटिविटी के साथ वो बच्चा अपने आप को तैयार कर पाए। तो ये एक बहुत इंपॉर्टेंट पार्ट है। दूसरा, ग्लोबल माइंडसेट भी बहुत इंपॉर्टेंट पार्ट है। वी मस्ट टू मेक अ स्टूडेंट ग्लोबली इंपॉर्टेंट। ग्लोबली इंपॉर्टेंट से मतलब ये कि ग्लोबल नीड्स को वो समझना चालू करे, क्योंकि आज से पहले ये पॉसिबल नहीं था। बट इन वर्चुअल वर्ल्ड दैट इज वेरी इजी। कहाँ क्या हो रहा है, किस तरीके से, हम लोग क्यों पीछे हैं? हमारे साथ प्रॉब्लम यही है कि जो रिसोर्सेज हैं वो रिसोर्सेज इंडस्ट्री में अवेलेबल हैं बट इंस्टीट्यूट में नहीं है। बट यदि हम उन चीजों को समझेंगे, अभी भी देखिए पब्लिकेशन उन्हीं पेपर्स के हो रहे हैं जिनमें कि नए सॉफ्टवेयर का यूज किया गया है, नए एप्लीकेशंस को यूज किया गया है, नए इंस्ट्रूमेंट्स को यूज किया गया है। वो क्यों? क्योंकि जब आप, ऑब्वियस सी बात है, कोई भी नए प्रिंटर से प्रिंटर निकालेंगे जो नए इरा का होगा तो वो और क्लियर आएगा। हमारी जो रिसर्च है वो ऑथेंटिक हो जाती है, हमारी रिसर्च की रिप्रोड्यूसिबिलिटी बढ़ जाती है, हमारी रिसर्च के जो एस्पेक्ट्स हैं वो जेन्युइनिटी की तरफ प्रूव्ड होते हैं। क्योंकि साइंटिफिक रिसर्च में कोई भी चीज आप बात नहीं कर सकते, साइंटिफिक रिसर्च में जो भी आप बोल रहे हैं दैट मस्ट बी प्रूवन। और यदि वो प्रूवन नहीं होगी तो मुझे नहीं लगता कि फिर उसको हम सही ढंग से कर पाएंगे। सो ओवरऑल मैं इससे ज्यादा आप सबको आपका समय नहीं लूंगा। मेरे जो एस्पेक्ट्स हैं वो सिंपल हैं। कॉलेज इज जस्ट फॉर मेकिंग लर्निंग अ डिसिप्लिन एंड बेसिक नॉलेज। बट जब हम मास्टर्स या पीएचडी में बच्चे जाते हैं तो उसके लिए हमें जो नर्चरिंग करनी है, जो हमें इनको बच्चों को प्रोत्साहित करना है, वो बैचलर्स से करें या 12th बेसिस से करें, ताकि वो वहां पर अपने माइंडसेट को पहले से उतना अच्छा करके आए और अपनी कैपेसिटी को बढ़ा पाए। एकेडमिक और इंडस्ट्री की जहां तक बात है, ऑब्वियसली बात है आपका इंस्टीट्यूट तो काफी अच्छा कर रहा है। They are interacting with them every day. I'm looking forward to you, and whatever the enthusiasm and the leadership quality in everyone. If somebody is giving a work to Geeta madam or Priyanka madam, is anyone, so they'll just take as a fight. No, we have to win the game, and we have to work like that. तो आप लोग अच्छा कर रहे हैं, मुझे लगता है, मेरी आपको शुभकामना है। आई जस्ट ग्रीट यू ऑल कि ऐसे ही आप अपने आप को ऐसा करते रहें जिससे कि जो हमारे बच्चे हैं, जो हमारे स्टूडेंट्स हैं, रिसर्चर्स हैं, इवन हमारी जो फार्मा फैटर्निटी है उसको लर्निंग मिलती रहेगी, उसको हेल्प मिलती रहेगी, उसको सपोर्ट मिलता रहेगा। सो इन एन ऑल, थैंक यू वेरी मच एवरीवन। डॉक्टर गौरव, थैंक्स फॉर इनवाइटिंग मी। डॉक्टर विकास सर, the president of Association of Registered Pharmacists, and they all are invited me to present some views. I'll definitely come with the next time. So whatever the slide I made for you all, I'll just deliver you. Okay. So thank you very much. Thank you. Have a great day.
Thank you very much, sir. First of all, a big thanks for collaborating with us for the organization of this virtual conference and your motivating words also. Every time when we meet each other, then you appreciate us, and that's means
No,
it
no, you
motivates us
people are
to.
really working well. You are working very well. Everyone. So I'll congratulate for a successful event even.
Thank you very much, sir. Now
Okay,
I
ma'am. May I leave,
request.
ma'am?
Yes.
Thank you.
Now I request Dr. Vikas Sharma, sir, President ARP, to please give vote of thanks.
Thank you, ma'am. Today I feel honored and privileged to get the opportunity to propose a vote of thanks on this virtual national conference. On behalf of ARP and behalf of organizing committee, we are deep regards and thanks to Chief Guest Dr. Misra sir, who give the time for this virtual conference. I am special thank to amenities speaker Dr. Vandana madam. He deliver a good lecture on the novel career for the cosmetology. He's a vast experience in the field of pharmacosmetology, and he give a very good lecture. Second, I am very thankful to Dr. O.P. Katare sir. सर को तो जितनी बार सुनो तो लगता है खत्म नहीं हो लेक्चर, सर का चलता रहे। सर का जो आध्यात्म और साइंस के साथ जो कोरिलेशन है, बार बार सागर की याद दिला देता है कि सागर में बैठ के कोई लेक्चर सुन रहे हो अपन। और सर ने जो काम लाइपोजोम पे किया और जो मेडिकल कॉन्वर्सेशन किया वो रियली बहुत एप्रिसिएटेड है। और आज के जो स्टूडेंट हैं, जो बच्चे हैं, उनके लिए ऐसा आइडियल उन्होंने सेट किया है कि लोग सोच सकते हैं कि इस तरीके से भी अपन पीजीआई के साथ कोलिब्रेट करके कुछ नए प्रोडक्ट उनको क्लिनिकल ट्रायल में ला सकते हैं। तो आई एम वेरी थैंकफुल टू कटारे सर। आई एम ऑल्सो वेरी थैंकफुल टू सुधीर सर साइंस के बाद जब बात प्रैक्टिकल आती है तो सर समझाते हैं कि किस तरीके से आज की चैलेंजेस को आप एक्सेप्ट करें और आगे बढ़ें। सर का अप्रोच रिसर्च को लेके और प्रॉब्लम को लेके थोड़ा अलग रहता है। आप सब लोग जानते हैं, हमेशा इंस्पायरेशन बोलते हैं सर कि अपन को ऐसा करना है, इस प्रॉब्लम को ऐसे सॉल्व कर लेंगे। ओवरऑल कॉन्फ्रेंस के मुझे लगता है कि जो अपना एम था वो फुलफिल हुआ। अपन ने ऐसे स्पीकर सेलेक्ट किए जिन्होंने एक अप्रोच जनरेट की कि किस तरीके से फार्मा में रिसर्च को अपन को आगे बढ़ाना चाहिए। Lastly, I sincere thanks to Dr. Kumar Gaurav sir, Principal Guru Gobind Singh College of Pharmacy, for organizing this informative conference. I am very thankful to Geeta madam, Priyanka madam, Rohit sir, and all faculty member, teaching, non-teaching staff who always directly, indirectly help to organize this virtual conference. I felt too proud and thank you to making this event is successful. कुमार सर, बहुत बहुत बधाई हो आपको, आपकी टीम को। अच्छा कॉन्फ्रेंस ऑर्गेनाइज की। स्पीकर और टॉपिक सिलेक्टिव थे, लोग समझ सकते हैं कि आगे कैसे रिसर्च करना है। जैसा कि कटारे सर और सुधीर सर ने कहा कि अगली बार वर्चुअली नहीं, अपन प्रेजेंटली मिलेंगे। मेरे बेस्ट विशेष आपके साथ, आप सबके साथ, सारे स्टूडेंट्स के साथ। एआरपी की तरफ से मैं आपको दोबारा पुनः धन्यवाद देता हूं, सारी टीम को जिन्होंने एक अच्छा ऑर्गेनाइजेशन किया। थैंक यू सर, थैंक यू मैम।
Thank you, Vikas sir, for your time and support. Thank you very much.
थैंक यू सर।
Thank you very much, all of you.